if you ask me, what's the theme for today? I'd say sperm matter a lot.
All right, so now let's talk about the guy who comes to see you. He's been on exogenous testosterone for 3 years. Presumably there are no sperm.
I would bet 95% confidence that he would have no sperm in his semen.
So what are you telling him now? How are you going to solve this problem? Oh, this is a very important point.
So 80% of conceptions naturally or at home occur when sex is front-loaded as opposed to reacting to ovulation.
Tell me about the impact of cold plunging and sauna and hot tubbing on fertility for men.
20 minutes on 104 degrees 3 times a week would probably make you zero.
What paternal age do you worry about?
National guidelines for sperm donation, 40 is considered older paternal age, 50 for sure, right?
Mm-hmm.
If you look at risks to offspring.
Tell me what your impression is of the effect of any or all of the above on fertility.
You double the stress on a man and his testosterone level will fall.
Yeah.
And then the sperm production falls for a whole different reason.
Hey everyone, welcome to the Drive Podcast. I'm your host, Peter Attia. Hey Paul, thank you so much for coming out to Austin.
Peter Attia, the man, the myth, the legend. I am so excited to be here.
So this is in many ways, I guess, what's going to be part one of a 2-part series I hope to do on fertility.
Yeah.
And given the complexity of it, I think the most logical way to do it would be to break it down into male fertility, female fertility, and there's obviously going to be things we talk about that'll overlap. But I was trying to think about the best way to start this, and the first thing that came to my mind was, can we just explain what's involved in conception, and maybe do it through the lens of the sperm? But like, how much of a challenge is this? I mean, obviously there's an enormous evolutionary pressure for this to go as easily as possible, but what is actually involved? So what happens for a sperm to fuse with an egg? What are all the things that are standing in its way, so to speak?
I mean, there are a lot of, so reproduction is an incredibly highly evolved million-year process and remarkably conserved among mammalian species, even among land species and water species of animals, vaginas, cervixes, uteruses. And the question is, why is it so much work for a sperm to get into the vagina, especially in, say, water, and then have to go through a cervix? And then the immune system in the uterus is very active because there's a hole in the woman to the peritoneum, to the abdomen. So it has to be highly protected. And then you have to go through the uterus. So there's a 10-inch, 12-inch swim, which is equivalent to about a 20-mile swim for a human.
Based on the size of the sperm.
After ejaculation.
Yeah.
And how much distance they have to go. And they do that in minutes, which is crazy. So it's an interesting challenge that nature has kept in place for a million years. And I really respect evolution. And it is why we're here for, eat, sleep, reproduce. So basically with ejaculation, the penis is shaped to fit into the cervix. So everyone wonders, is it getting to the right spot? It's also interesting that the semen is coagulated and then it liquefies. And that's because there's a lot of species of lower phyla that they have to leave as soon as they have sex. Otherwise they'll get killed, like praying mantises and black widow spiders. So you got to get out of there as a guy. So our ejaculates in humans are sticky.
By the way, is there an evolutionary explanation for that phenomenon?
I have no idea, Peter. I have no idea why you would do that. I don't know why one queen bee and the bees in the hive die after mating. I have no idea why that's an advantage. But I guess females are prioritized in evolution, and that makes sense. So the anatomy is perfectly defined. So a lot of men think they're having trouble placing things, but that I usually don't worry about it because the cervix and the penis, it expands, it forms a seal. Then there's a crypt. Sperm have to go through a crypt. A channel, which is only a few sperm make it. So 100 million sperm may start out, maybe 5 million make it through the first barrier, which is the cervical barrier. The vaginal fluid is acidic.
How acidic?
5,
Okay.
pH of 5, and the semen is a pH of 7. It's all buffered. So as soon as that's as a hostile environment, so it has to get out of there quickly. As soon as it liquefies, there's sugars in there, and then they go through the cervical path. So 5 million will make it. 1 out of 20 1 out of 20 makes it through the cervix. Then 100 make it to the fallopian tube, and then 1 will make it to the egg.
Literally only 100?
Right. And this Setlage studies in the 50s where he had women have sex before hysterectomies. And then he swabbed different parts of the reproductive tract. These are young women for different reasons, not infertility, and found these numbers. And that's the basis for our move to technology from 5 million moving sperm is when we start doing inseminations versus sex, et cetera. So those are based on the kind of numbers of sperm that reach the uterus and reach the thing. What's really interesting is there's some fascinating research. Everyone thought the Vanguard sperm wins.
Yeah.
Right. So it's the Phelps sperm that's going to make it.
Right.
And there's a company out of Boston called Eric's Biosciences, and I'm consulting with them, disclosure. But they've discovered that sperm work in phalanxes. So because the immune system is so vibrant in the uterus, the first round of sperm gets through the cervix and typically absorbs the immune system. It secretes FcR receptor.
And by the way, we've referred to the immune system a couple of times now. What is it? Is it just a bunch of antibodies? Are they B cells? What is
Everything.
The barrier?
There's T cells, B cells, and antibodies.
It's a full immune response.
Absolutely.
So it's a specific immune response. It's
anything foreign.
Wow.
I mean, if you think about it, and there's also a mucus plug that exists for 28 days a month to prevent anything from going through because it's a hole into the woman's body, and peritonitis is severe, right? So the cervical mucus thins, and that's to let sperm through for 2 days a month. It's incredibly detailed, perfectly orchestrated system. So it looks like the first round of sperm get through the cervix, get into the uterus, and they get demolished like a phalanx, like a Roman phalanx. And maybe a second round goes through, and they get demolished, and they're secreting the FcR receptor in the immunoglobulin, because that's what antibodies bind to. So the female antibodies bind to that, and we don't know how many phalanxes go through, but then it's like a run up the middle, and then eventually a couple of sperm or fourth make it, and the immune system's deactivated, and they get there. It's wild. That's a whole new way, and that can be measured now. And there's actually going to be an assay available to look at whether you're doing this, and they're calling it a sperm cycle, almost like ovulation, spermulation. But it's an 1.5-hour cycle when the phalanx is working, sperm are deactivating the immune system, and then maybe they don't. So there are groups of there are ejaculates, which is a group of sperm, some of which do this well and some of which don't. And that can be a whole reason for infertility. If you're not able to deactivate the system, you're not going to be able to get through because the immune system is active.
Let's go through those numbers one more time. About 100 million ejaculated
at the cervix.
Yep. 5 million get through
to cervix into the uterus.
Yep.
100 to 500 get to the fallopian tube, and 1 gets to the egg.
Wow.
So the question is why do you need so many sperm? The classic answer I used to give is they don't like to ask for directions. Men don't like to ask for directions, but this is probably why.
Let's also define what makes up the ejaculate, because we've talked about the sperm. So how are sperm made? Because an important consideration for a sperm is it can only have half the genetic information contained within all the other cells in the man's body. So when does that take place?
So the testicle makes sperm. It takes about 60 to 70 days. And it's a process called meiosis. So in a car assembly line, you want everything to be the Model T assembly, mass produced. You want it all to be the same. In meiosis, which is unlike mitosis, you want things to be different and to be a little easy peasy. So you get what's called recombination. And so that's the source of evolution. So the genes, the chromosomes blend in a different way and separate a different way. And through that process of a couple of those, you get half the number of chromosomes, which is required to join the other half.
But it's not always the same half.
Correct.
Yep.
And it's loosey-goosey. It's not the same as when it started.
Yeah. A funny story, which I think I've shared on the podcast before, but if not, I'm sure someone will be amused by my stupidity. I had to take the MCAT before doing any of the pre-med stuff because I had studied engineering and then decided I wanted to do medicine, but didn't want to spend 2 years preparing, 1 year taking the post-bac and then the MCAT and then doing it. So I was like, I'm going to just wing it and take this MCAT having never taken a biology class since high school. And I think I took I took freshman biology, right? So I am studying my heart out for this little MCAT test, and the physics and the chemistry are fine, but this biology thing is killing me. I bought this cheap study guide. I didn't have the money, or I didn't want to splurge for the official study guide. Like there's an official MCAT study guide. But the thing was like $60, but there were these knockoff books for $10. I was like, they're just as thick. So I buy one of those. And every time I encounter the word meiosis and mitosis, I assume it's a spelling mistake because I bought the knockoff book. So I'm treating it as the same thing. Every time I see the word meiosis, I'm like, these guys, they just misspelled it, idiots. It's mitosis, mitosis, mitosis. Finally, on the night before the exam, which I still remember, it was August 17th was the day I took the test, I realized they were two totally different things.
Big things.
There's a big difference. God. That realization might have got me into med school because, I think I barely got a 10 on the biology section, which was, it's hard to get into a good med school if you get below that. So anyway, to this day, I get such a chuckle out of the confusion of the nomenclature. But again, just to explain for people, mitosis is what happens when cells are dividing in our body constantly, where they're trying to create a perfect replica of the entire suite of DNA. So really, to my knowledge, the only time we're undergoing meiosis is in the creation of an egg or a sperm.
That's right.
Okay. Now, remind me, are women born with all of their eggs? I feel like that's something I vaguely remember.
You'll have a guest talk. Yes, 5 million eggs at birth.
Okay.
5 million eggs at conception, 1 million eggs at birth, and you basically ovulate 1,000 in your lifetime.
Okay.
So by the time you're 45, you're out of eggs. You actually ovulate 1 a month, but you actually produce 10 a month.
Got it.
So you lose 10
for every one.
Right. So there's a lot of waste, but they're stuck in a stage of perpetual space where they're just boom, and they get older and they don't evolve really. And then they mature when they're asked to at that time, but sperm are constantly renewed. So if you look at
Is that just a mass space problem because the testes are, if we did the same thing women did, would we just have to have an enormous set of testes? Is that the reason that we continue
about that, Peter? Why do you think out of the box like that? So no, I'm not sure.
Okay.
There's a whole issue of, what's the source of human evolution? It's really sperm.
Yeah.
Because they're constantly dividing. They're constantly influenced by the environment. And they're throwing off mutations and epigenetic changes. And what's most interesting for me for this talk is that whatever happens in sperm happens to offspring.
That's an interesting point, right?
So it's transgenerational.
Yeah.
So
does that mean that the father is more likely to pass on environmental stressors than the mother?
Probably, yeah. And that's definitely been shown.
Okay, so let's go back to it. So the sperm is the actual cell. Where does it get the little tail from, and what is the other part of the cocktail that is?
So one of the most magnificent transformations of a cell in the body is the making of a sperm. It starts with a spermatogonial stem cell, which looks like other cells. That spermatogonial stem cell is actually the first and the bottom of a tube. There's 12 stages of spermatogenesis. That cell is remarkable. It's actually the human male embryonic stem cell. So I have a patent on that cell. Because if you take that cell and you put it in a niche environment like an embryonic stem cell, it'll become embryonic almost like, and it can become multipotent.
It's pluripotent?
Yes. Multipotent, we don't know about pluripotent, but you can form tumors and you can form bone, mesoderm, ectoderm, and endoderm. You can do all 3 layers of the body with that adult spermatogonial stem cell.
Is there any other cell in the body that is capable of that?
No. There are stem cells in the bone marrow. There might be stem cells in fat, but we showed that the capability of the cell is magnificent. And so I think it's the source, like women have eggs and embryonic stem cells. That's the male embryonic stem cell, in my opinion. It hasn't been taken advantage of yet with cell-based therapy, but it is really incredible what this cell can do.
And a man potentially has access to this cell his entire life?
Yep, as long as he's making sperm. So that starts out, and it usually reproduces mitotically, and then at puberty, it'll go down the path of meiosis, which is a couple steps more than is mitosis involved with meiosis, but it's the having and the mixing up of the chromosomes and the newness of the genome introduces mutations and stuff. And most mutations are bad, and some are good. And you don't really think about that. But we could have a long talk about genetics versus epigenetics.
Actually, let's focus on that for a second. I hadn't considered that. So when the cell that is becoming a sperm undergoes meiosis and it divides, what's the fraction of times when this becomes an aneuploidic sperm? And explain maybe to people what aneuploidy is, and what's the process by which that thing gets discarded?
So if you look at healthy human sperm for chromosomal content and what's correct and what's incorrect, probably 2% of them are off. They're still being made. They're just off because it doesn't really click the system to negate it. We don't know at what level of chromosomal abnormalities the system will say this is a bad
absolutely
product,
defective. Yeah.
Right? But I would say if you look at making of sperm, it's very logarithmic. You're probably looking at 1 out of 4 that are being made go through the epididymis, which is the next 10 days, which is a collecting duct after the testicle where it matures, gets epigenetically modified, and you'll see these zomes, different epididymosomes and things like that happening. And there's a lot of post-production modification, not of DNA essentially, but I think there's a filter going on where a lot of the bad aneuploidy comes out, because if you look at the chromosomal abnormality rate in testicular sperm before it goes through the rest of the system and compared to ejaculate, it's higher. It's 2 to 3-fold higher.
So something is getting filtered.
So there's a filter.
Yeah, and just so folks know, when we say aneuploidy, we mean you don't have one copy of each chromosome. You either have none or you have 2, or anything that's not 1 is bad.
Right.
Yeah. And when aneuploidy occurs in the fusion of the sperm and the egg, do we know, I guess we can figure out pretty easily if it's maternal or paternal in origin?
If you look at the embryo, it's kind of hard to tell. There are some markers of paternal and maternal origin. It depends on where you're going back in mitosis and meiosis. So they can sort of ascribe it in the embryo. In the sperm, you're really going to have to look at the sperm. And if you see a translocation, some characteristic change in sperm, and you see it in the embryo, then you know it's paternal.
Yep. Okay.
But not usually. And 98% of sperm are typically normal. And a guy with infertility, it might be 95%. Here's an example. If you have a patient with Klinefelter syndrome, a male with an extra X chromosome in every cell in their body, or a transgenic model with that feature.
So this is a man who's XXY instead of XY.
Right, so 47 chromosomes, not 46.
Yep, and phenotypically, he kind of has a distinctive look, right?
10% of the time.
Oh, really?
Yeah.
90% of the time,
a
man with Klinefelters, you'd never know.
Yeah.
Okay.
So that's the board question.
That's the board question, right?
The MCAT question.
Yep.
So all right. So in these men, if you look at their sperm aneuploidy, right? So every cell in their body and in the mice, and the transgenic mice, all have an extra X chromosome.
Yep.
Only about 10% will have it in the sperm.
Meaning they will produce an X or a Y sperm.
Just like everyone else.
The only difference is they have a 2/3 chance of producing an X and a 1/3 chance of producing a Y, I'm assuming, instead of 50-50.
Don't think we know that. That's math, Peter. That's math. Yeah. So biology is not math, remember? So and I had 2 Klinefelter patients yesterday that I operated on, and they're not doing pre-implantation genetic diagnosis of the embryos that they're going to create from their sperm because the chance is not that high. So it goes from, in mice, 0.1% chance of normal men having an XXY sperm or an aneuploid sperm, an abnormal sperm, to 1%. In humans, it goes from 1% or so to 10%.
Mm-hmm.
But 90% are still, that's remarkable.
That's amazing.
It's remarkable how efficient this is.
I interrupted you. You were in the process of explaining how we actually make the sperm, where the tail comes from, and the whole process.
It's all done in the testicle.
Yep.
It's an amazing machine. The weird question we should maybe get to is, why is it out in the breeze like that?
I assumed it was temperature related.
But why? Why
does it need to be a little cooler?
Yeah. Why? We've been asking ourselves in the field for years.
I mean, I would guess, because I like to make things up, that it's so energetically demanding that it's giving off more heat in the process of creating something that is going to be so efficient to be able to swim 20 miles effectively. And that's very glycolytic, I'm assuming, right? Like the amount of ATP that must be generated.
75 mitochondria per sperm. That's like, it's like an electric motor on each wheel.
Yeah. So that would be my guess. Is you
it's an overheating problem if you tried to keep those guys inside. And I think overheating could be translated to oxidative stress, which is a cause of a lot of infertility. Anyway,
we don't know is the answer.
Right. And it is interesting that, ovaries are inside. So men get in hot baths and they're cooked. Women can get in hot baths and they're okay. So it's
funny. I had a buddy over who shall remain nameless, and he does not have kids, but he would like to have kids. And we sauna'd the other day, and he went up to my freezer beforehand. I didn't really know what he was doing in there. I thought he was getting a drink or something, and he came down with ice packs. And
there's two companies that do that.
Yeah. So he basically, we were sitting in the sauna, and he was in the sauna, but he's got ice packs all over his groin, and I immediately understood why he was doing that. But yeah. We'll come back to whether or not that's an important strategy for men in saunas who want to have kids.
Right. So the complete sperm, the first half is called spermiogenesis. Spermatogenesis is the whole process. Spermiogenesis is when you go from the round cell stage and you get half the number of chromosomes, and then you have to make a tail and then a whole motor assembly. And that is the most profound transformation of a cell in the body. It takes about 3 weeks to go from that stage. And we're learning now a lot of it's vitamin A driven.
3 weeks?
Of the 6 or 7 to make a sperm. And then then it's complete and non-modal, and it's packaged. It has
Give us some size comparison. Before the tail is on, what is the size of that cell?
Probably similar to half the size of a lymphocyte.
Okay.
Or half the size of a red blood cell, a couple microns.
And then the tail makes it
35 micron tail, yeah. So really magnificent engineering feat. It's got microtubules in the middle, and there's these sort of links to the tail. It's like a kite, and the engine runs it, and the tail wags. And it's remarkable. 300 genes control movement of sperm alone. There's mitochondrial DNA in there, all that stuff. It's just wildly compact, 10 times more compact than any other cell in the body.
From a mitochondrial density standpoint?
From a cytoplasmic standpoint and nuclear standpoint, it goes from histones to protamines. So the DNA is condensed a lot more because it's got to go on the road, right? So it's got to be packaged really well to survive outside the body and be in good shape because it's the transgenerational. So a lot of energy in that. And then during the epididymis, which is a collecting duct, which most people don't know about.
Oh, sorry, one other question. Where is the ATP or carbohydrate or whatever, the glucose stored in the sperm?
Stored probably in the cytoplasm and in the tail.
Okay. And it's interesting. It's like when you think about a rocket ship with its payload, it uses a solid fuel, obviously, as opposed to a liquid fuel. It's packaged for that one shot. I assume it's the same here. Yep. There's no transporters to bring glucose in or anything. No. It has its solid fuel, one shot, go for broke.
That's right.
Yeah, it is a lot like a rocket, isn't it?
It is a lot. So that's going to bring the physics in, Peter's physics. So then there's 2-week period where it stays in the epididymis, which is basically a 35-foot tubule with estrogen, and there's a lot of post-modification of the sperm.
The epididymis is 35 feet if you stretched it out?
Roughly, yeah.
And just for folks listening,
700 feet of tubules in the testis.
The epididymis is on the back of the testes.
It's a comma-shaped organ in the back,
yeah. And this, we'll come to this, I'm sure, later. This is prone to infection, and that'll probably factor into maybe some of the issues that deal with fertility.
Right.
Yeah, okay.
So epididymis has been relatively understudied, but it has actually become very important. Epididymisomes, and there's a lot of modifications we don't really understand. I wrote the chapter for our textbook on reproductive physiology, and it really is a lot of work in the 50s and 60s, but now we're beginning to understand like DNA fragmentation and the quality of sperm is driven by the epididymis. A lot of the quality of sperm, not the shape and stuff like that. But two things that happen.
Based on its residence time within the epididymis?
And what other environmental influences that occur there, because the epididymis is not as walled off from the body as the testis is, immunologically and otherwise. It's more susceptible to drugs, exposures, heat, etc. Testis is very walled off. Very little happens in the testis because the Sertoli cells that line the tubules have a blood-brain barrier, blood-testis barrier. Same as the brain. It's highly protective.
It's as protective?
Yes. Harvey Cushing at Yale did that in the late 18th century, took brain-dead patients, injected them with dye, methylene blue, I think, and the blood-brain barrier came about when nothing went into the brain and nothing went into the testicle. Those are the two areas of the body that were completely immune from normal transport processes.
Wow.
Blood-testis barrier. So The two things that we know happen in the epididymis after production of sperm are motility improves. So sperm begin to learn progressive motility. So they start moving forward as opposed to not moving or moving in circles, which is important. And the most curious thing is they learn how to smell.
And meaning there's basically a chemical signal that they need to be able to track to, which I'm assuming is the egg.
Mm-hmm. So the follicular fluid. So they actually detect follicular fluid. So if you take testicular sperm and inseminate it into a uterus with insemination technology, it'll just be killed. If you take an epididymal sperm and you do that from the top of the epididymis, it'll maybe run in circles and it'll be killed by the immune system of the female.
It has to go through that whole epididymal cycle.
Once it's at the end of the epididymis where it's stored.
And that's how many weeks?
10 to 14 days. 600 million sperm live in a bucket, a pot of soup called the cauda epididymis, and you ejaculate from that pot, which tells you a lot about sperm quality because it can get old. But that sperm, if you put it in it, will know exactly where to go and it'll move forward because it's like a shark sensing blood in the water. 1 part per billion of follicular fluid can be sensed by a sperm. So that's incredible. So it is literally an olfactory sense. It's a smell sense that sperm have for follicular fluid. So they know exactly where to go.
Again, you have to wonder how many years it took to perfect this system, right?
You do have to wonder.
Do we know what the chemoreceptor is?
Yes, it was published in Nature recently and stuff like that. So it's really interesting. So it's an olfactory type receptor. Yeah.
Do we have anything else that we can? Is there any
smell, Peter?
Yeah. Is there anything like what is the most noxious thing that we can smell with our nose, and at what concentration can we detect it?
I have no idea.
Yeah, because I wonder, just for comparison.
Give me a minute.
Yeah. No, I've always thought about this. If I like to hunt, and so anyone who's ever bow hunted especially knows that animals can smell at a level that we can't even fathom, right? Like they can smell us literally a mile away if the wind is just blowing their direction. So it's always seemed to me like we have really, really insufficient noses, right? We were given lots of superpowers in many ways, but smell wasn't really one of them.
I think I would agree with that. And it'd also say that if you block a sensory bank of the 5, others increase remarkably, like Braille. I mean, I'm a microsurgeon. This stuff matters a lot, but I can't do Braille or hearing. I think you can crank it up if you lose a sense. And you see that with people who are deaf. I mean, your ability to see, and I don't think seeing better is really the issue, but hearing and smell, I think it can crank up.
So 1 part per billion, this is kind of remarkable. You mentioned that at the base of the epididymis is basically the launch pad.
Mm-hmm.
How many are stored in that?
Half a billion.
500 million. So 5 ejaculations.
Yep.
Okay.
So it's a pot.
And what's the time to rebuild that?
You mean to
what's
fill it again?
What's the rate at which you fill?
Oh, I don't know. And the rate at
the
which you
reason
draw
I ask is if we're going to come to this, I'm sure, but is there a frequency of ejaculation that is too much, that if a guy is, say, ejaculating every single day, is that insufficient to get a complete replenishment, where if he's having infertility, you would say you got to move it to every other day or whatever the number is?
So that's a great extrapolation of the pot of soup idea. And so on that note, I would say typically we recommend every 2 days of abstinence between sex every other day.
Is it to optimize?
Right, but not for the semen analysis. That's for conception.
Okay.
Because it depends how old you are in your biology, but most men need a day or two to refill, to recharge completely, a day or two. So that's why we do it. That's why we recommend that. And that's sort of a generalization. Some men are fine every day. Right?
Okay.
I had a guy once who had to bank sperm for hepatitis treatment. And he looked like Mickey Rourke and had a wooden leg, and he's about 50. And I said, you're going to need to abstain for a couple 3 days to do this semen analysis so we get a good sample. I want it to be, optimized one. And he looked at his partner and he goes, and she looked at me, she goes, she grabs him and says, you can't do that. He's every day. You can't do that. I don't know what he's going to do. So he's like, he was panicking that he had to hold off for a day. I said, how often do you have sex? He said, twice a day, every day. I'm like, okay. So that was great. And then I had one man, wonderful orthopedic surgeon at Stanford, and I asked him on my questionnaire, I said, how often do you have sex? And he wrote 0.00001356. Which meant
he divided once a year.
Well, 0.00 weekly, 0.00155 for Avogadro's number, right? And I'm like, which meant he was so frustrated that it was a beautiful way to say that. It was the 0.001355.
Yeah.
So anyway, so there for a semen analysis for diagnostics for infertility, when you abstain longer, your sperm count will rise, but your motility will fall because it's older. So there's a
min-max curve that you're optimizing for,
right,
which you would say is
3 days would be about right. When you're not going to gain that much, you're not going to lose that much motility. After that, it starts going. So there's sort of biological variability, which we try to minimize when we do the semen analysis. So 2 to 4 days of abstinence. That's a different period than what we're recommending for sex, which is every other day.
Mm-hmm.
And that's based on New England Journal paper where they looked at, I think 700 couples, and they had them keep diaries. I think it was a Boston-based paper, keep diaries of how they had sex, when they ovulated, and when they got pregnant. And then they said, do what you normally do, and then give us the diaries. And then they evaluated them, and they found that having sex, say ovulation is day 15 of the cycle, when they started having sex on 9, 11, 13, there were significant pregnancy rates, and every other day was the optimal interval. But even 5 days before and 3 days before, there were substantial pregnancy rates before ovulation. But if you waited to ovulation and then had sex, that's about 20% of conception. So when you get the kit, don't react to it. Predict in front of it. So front load the sex. Very important point.
And why is that? Is that because
there's a reservoir effect in this uterus? It's managed. Sperm survive for a day or two. So when men are under stress.
If ovulation is day 15, how could a day 11 sperm survive 4 days?
Because it's nurtured once it's past the vagina.
But how many of them are surviving? Is it literally the lone wolf, or
Probably.
Is it the last 100?
Maybe. Some of the sperm bind to the oviduct and wait.
Remind me where the oviduct is?
So there's the uterus, and there's the fallopian tubes.
Fallopian tubes.
And that's the oviduct. And
the oviduct is right below where the ovaries
fallopian
sit.
tube, essentially. Yeah, they bind to the endothelium and just park. If there's no egg, they'll just sit there and they'll bind.
Again, going back to our moon analogy, this is after you've done stage 1, stage 2, stage 3. You're now out of gravity, right? Like it's actually not an energetics problem anymore.
Right.
You're not fighting.
Death star problem, right?
That's right. You've escaped the hostile environment, in this case of gravity.
So now it's a fun place. It's the right pH. It's warm.
So do we have a sense, this would be a very interesting experiment, of what is the longest duration that a sperm could survive for conception? In other words, to do the experiment, let's just make it as a thought experiment. You had, a large number of women that you knew were going to ovulate on day 15, and then you would have them, have intercourse on day 7, 8, 9, 10, and you create a histoplot or a distribution of what's the frequency of pregnancy across those things, and kind of ask, like, what's the bottom 5th percentile, which is the theoretical possibility.
Yeah, that's a good one.
And then the same thing after, right? You want to develop the bell curve of the whole thing.
Well, we know that once the egg is ovulated, about 8 hours, and then it's over.
Oh, this is a very important point. It really needs to be front. If it's only 8 hours of survival, this is...
After ovulation, about 8 hours, it's dead if it's not...
This is a very, very left tail curve.
Correct.
Ah, I did not know that.
So you want the sperm there ahead of time. So 80% of conceptions naturally or at home occur when sex is front-loaded as opposed to reacting to ovulation. And most of the apps that are available nowadays will tell you that. Peter, you're drawing a graph.
I am. I have to draw.
And it looks like it's algebraic.
This is incredible. Okay, yeah, it's easier for me to think about these things graphically. And to think that it basically shuts off at about 8 hours.
So I give some more physiology. There was a study that showed how long it took to make a sperm. And it was published in Science, I think, in the 60s, and they gave men tritiated water. They gave men radioactive hydrogen, tritiated water, and then they biopsied their testicles, which could never be done nowadays. But I did it a little different. I gave deuterated water with a group at Berkeley, and we gave healthy men deuterated water for a week, and then we checked the first.
Sorry, dumb question. Why didn't they just measure the ejaculate? Why did they have to biopsy the testes?
Sure. Why did they just want to know about spermatogenesis? But we didn't want to biopsy
They actually
testes.
wanted to torture the guys, but.
And they weren't, I don't know that's wild, but that was the best data. And we did deuterated water, which is not radioactive, and we could measure that. So we gave them a dose, and then we watched their ejaculates weekly.
Yep.
And we looked for when the hydrogen showed up in the DNA. And it was an average of 74 days. So normally say 3 months to make a sperm. So some women were 42 days. And that's going through the epididymis and getting ejaculated. So that's so we talked about maybe 2 months in the testis and 2 weeks, a week or two in the epididymis, and then maybe a couple of weeks to ejaculate. And this was all the average 74 days. So it actually changed the timeline enormously to a much faster one. So 74 days. So when you do anything to a man fertility wise, you're not going to expect to see anything change for at least 2.5 months. And when you talk about full replacement of that semen, it's probably end up being 90 days when it's all replaced. The pot is replaced.
Yep.
So that's a limitation of what we do, because 42-year-old women want now, and we have 3 to 6 months. When I did a study on fixing varicoceles, which is an infertility problem in men, it's surgery, and I looked at the mean time to conception, it was about 7 months after repair, which is 2 cycles of sperm production.
So let's now define infertility. Again, we've been using this term quite a bit, but I suspect it actually has a formal definition.
It's 1 year of inability to conceive after sex, using sex.
Okay. So
doesn't have to be timed intercourse, just has to be whatever the couple does when they think they're trying to conceive.
Okay. When someone shows up in your office, is it usually after they've gone down the rabbit hole of troubleshooting the female partner, or are people doing this in parallel?
There's a large bias in Western worlds about how infertility is evaluated, and it's a complex, the reasons are complex. But I would say my practice is not typical. So most of my patients have been through a lot before they come to me. And typically, I think Keith Jarvie's data was good, and about 23% of men get a formal evaluation for infertility before couples go through IVF in North America.
And how does that differ from the rest of the world? Let's say from Europe?
I don't think it's been studied in the rest of the world. But there are countries like Germany and Spain with single insurers and government pays. And it's also recommended by society guidelines like American Society of Reproductive Medicine, WHO, et cetera, that both partners get evaluated simultaneously. But the bias is female gets very evaluated for lots of money, and the men typically may get a semen analysis, but may not. And it's very complex reasoning there. It's a different beast. They're not part of the problem. They refuse to do it. There's a lot of denial. It does get at your masculinity a little bit to get checked out and things. So it does go deep for men. It can be a little bit of a problem. So I would say that lately with large insurers coming in, Progeny, Maven, and things like that, you're seeing a lot more men up front, which is fabulous. And we can have long discussions about the biomarker concept, why that's good for the field and good for men's health and good for longevity.
Okay, so let's talk about your workup. What do you do when a guy comes in, and what are the things you want to know about him?
So getting a guy in is great. Usually they're dragged in by their partners. Usually the partners come along to make sure they show up. For me, it's one visit. So we do one visit and I do everything else where they are. I don't ask them to come in a million times anymore. So it's a very different kind of practice. But I try to get everything done in one visit because when you get them there, it's rare to get them there. And the physical exam, so you do a history, a very thorough history, which is usually preceded by a questionnaire. I give like 200 questions, and that has all the hot bath stuff and all the exposures they have. And they have to do that before they see me, because that's a really important part of it. If you could pick one in a multiple choice question, what matters the most is probably the history. History of paternity matters. A history of exposures matters, et cetera. Physical exam, very important. 1 to 5% of male infertility can be due to a major medical issue, testis cancer, diabetes, things like that. So physical exam, varicoceles is very important. You could be missing a vas deferens. 1 in 500 men have perfectly normal testicles, but they have a natural vasectomy. It's congenital absence of the vas. They're going to be sterile or infertile.
Can you explain we haven't talked about how a vasectomy works and why a guy still ejaculates but is infertile. Explain what the vas deferens, how the whole thing works in the plumbing.
Right. So we didn't answer that question, which was what's the semen consist of?
Yep.
So it's about 10% vasal fluid with sperm. It's about 80% seminal vesicle fluid, which is an accessory sex gland in the back with a prostate, and about 10% prostate. So typically during ejaculation, prostatic fluid, which is clear and sticky, will grease the barrel of the urethra, sort of pre-cum. Then during the ejaculation process, the pellet of sperm gets pumped from the vas deferens into a chamber called the ejaculatory duct. And this happens quickly. And then the seminal vesicle, which is like a bladder, contracts, sends it into the prostatic urethra between the bladder and the outside world. There's 2 valves. One is the bladder neck, it closes, and one is the urethral sphincter that we pee through, and that opens, and it gets forced out with muscular contractions. So that happens in seconds.
Yeah. So therefore, if the vas deferens is clipped, you're getting essentially 90% of the volume. You're just missing the 10% of the volume that contains the payload.
So in 3,000 men I've done vasectomies on in 30 years, 2 men have said, my volume went down. And I said, really? One of them banked sperm, and he had a semen analysis before and after, and he did go down by 15%. And he noticed it, and I said, good for you. What do you want to do now? But so it can be noticeable.
Yep.
But not usually. And so the color is the same, the opacity is the same, the whole process of liquefaction is the same, viscosity, et cetera.
Got it. So physical exam, do you need an ultrasound? How are you able to detect if a person is congenitally missing a vas deferens?
It's just pure physical exam.
What you feel it?
Mm-hmm.
Interesting.
So my fingers can feel 2.5 millimeters. And the vas deferens is like a piano wire. I mean, it is different than anything else in the cord. So I did a study. A third of my men with absent vas were only found out having procedures until I saw them. I usually just do the exam, but it is an expertise thing.
Yeah. It's not like the PCP can figure this out. You have to be doing this all day, every day.
I think you need to be trained on that.
Yeah.
But if you're well trained, it should be purely a physical exam.
What percentage of men are congenitally missing their vas?
1 in 500.
1 in 500. Okay.
The most common genetic disease in America is cystic fibrosis. So the big implication is these men can't conceive naturally. They have a natural vasectomy. We use sperm retrieval techniques in IVF, but they definitely have the chance of passing on cystic fibrosis to a child.
Why is that?
Because it's a very interesting biology, but men with cystic fibrosis, the most common genetic disease in America, have no vas deferens.
Okay, so what's the Venn diagram of cystic fibrosis and congenital lacking vas deferens?
The genes for that were discovered. It's chromosome 7. There's 1,800 mutations, maybe 2,000. So they cloned the genes and got the variants in the late 80s. And then they found there's another group of men who are perfectly healthy, do not have cystic fibrosis, which is a major metabolic disease with a short life expectancy, better now. Those men had absent vas deferens in the absence of disease. They took the gene sets and looked at them, and they were the same. Just not as many, right? So you have homozygous or heterozygous. So you have a carrier for cystic fibrosis will have an absent vas, but a full-blown CF patient, cystic fibrosis patient, will have no vas deferens too. So it's a form fruste of cystic fibrosis, but it doesn't have all the chemical and metabolic side effects.
So in other words, when you identify a man who does not have CF with a congenitally absent vas, there's a very good probability he's a carrier of CF.
Yes, and you can usually define it.
Which we can genetically
80%.
test easily. Okay.
Yeah, and then you have to worry if there's a 4% chance in America anyway that a partner might carry it.
Yep.
There are 2 carriers. You have a 1 in 4 chance of having a very affected child.
Yep.
So that's the big concern in my practice. And I'm proud to say in 30 years, we have no CF children.
Okay.
It's all about good engineering and doing it right. So that's the vas deferens part.
What else on physical exam are you looking for?
So cancers, infections, epididymitis.
Yeah, tell me about epididymitis. Obviously, anything that interferes with that section of the journey is going to be critical. And remind me, is it EBV that we typically, or measles, mumps? What's the infection?
Mumps.
Mumps, okay.
But that's typically, So among viruses in the world, there aren't many that get into the testicle like other things. Very little gets into the testicle, similar to the brain. But the mumps virus does it about a third of the time when you're a child with mumps. The parotid gland infection. It's a glandular disease.
Yep.
So it really only matters when you're pubertal and you get mumps. And then it goes to lots of glands. It go to your pancreas and cause diabetes. It can go to the salivary glands and then go to the testicles. It's some kind of, there's an open time, open.
So just one more reason why everyone should really get the MMR vaccine when they're a child is like notwithstanding the tragedy
this is a big deal,
of children dying from preventable diseases. But this is another non-lethal but significant complication of the mumps.
Absolutely. And it will cause viral necrosis and edema of the testis. And similar to a brain, it's in a calvarium, right? The brain is in a fixed space. So when it swells, you got to do something because it can die if it swells too much. Testicle is a fixed cavity with the tunic albuginea. And so if it swells too much, it necroses. And then you get fibrosis. And then you get sterility. And it's, I've got techniques where I can find sperm in lots of these men, very little pockets, but most of it, you're ablating the testis. It's going to scar and die from ischemic necrosis. So Zika, Ebola, the CDC called me when these were coming around. Zika's been transmitted through semen. It causes the anencephaly issues with child. And when these pandemics recurring, Ebola too. I got a call that there was an Ebola patient who survived, went to the institute, survived the hemorrhagic fever, and then a year later transmitted Ebola to a partner who transmitted to 6 other men. And it was another outbreak in South Africa.
Meaning the patient that survived Ebola, the virus managed to survive in the testes?
Somewhere.
Okay.
But it was transmitted sexually a year later when he was well.
When he was asymptomatic. He had already developed immunity.
Right. So we don't know about testis, but we know that mumps will do that to the testis, but Zika is also persistent in the semen.
But in that case, you have to think they would wherever the virus hung out, it had to be very, very immune privileged.
Yep.
Huh.
Or at low levels, like low viral loads where there's no disease. I'm not sure. But these are concerning cases.
But then it was transmitted at a low viral load, an even lower viral load.
Right. So it's tricky.
Yeah.
And there may be bulbourethral glands, maybe it's seminal vesicle. It's hard to know, but it's getting away. So, but most viruses don't go there. So the big one would be COVID, right? What did COVID? There was a big deal about the AC receptor being in the lung and being in the testicle, and maybe COVID infection would make you sterile. There was one Zika paper in Nature that looked at if you infect mice, or was it rats with Zika, the testicles shrivel up and they get infertile. And that caused a huge scare in the field, but we really didn't see it. We maybe see it we don't see it in fertility.
And what is it about the Zika virus that does this?
We're not sure why it did it in rats. But it's a blood testis barrier thing. It's an amazing barrier and nothing really gets through, including viruses, but mumps does only at puberty.
And Zika does.
Zika does in animals, but we didn't see it in humans.
But I thought you said that Zika was leading to anencephaly in cases.
Yeah, but that could be seminal. That could be just in the semen itself, not in the sperm. Like Ebola is probably seminal, not testicular. It's not on sperm. It's around sperm or in the fluid. That's the conclusion so far. So COVID, the big worry was when this Chinese paper came out, like, oh my God, it's going to the lung, binding to the AC receptor. Its testicle has it too. It's going to make men sterile forever. And there were cases of infertility with bad infections. And, was that just the fever, which typically does it even after a flu, or was that COVID specific? And we didn't know. A couple of colleagues did some papers. One, which impressed me, was out of Cedars, was a bunch of men, maybe not reproductive age, died with florid COVID. So they got autopsies and they looked for virus in different locations in the body. And I think out of 10 men or 7 men, 1 had it in the testicle. So these are men with the worst infection ever, the highest viral load you can imagine, and only 1 of them had it. So I believe that there is a risk of it. But I'd say in the 1,000 men I've seen since COVID, I think there were 2 cases that I would say were unexplained, where men were either fertile or had normal semen quality, had a bad COVID infection, maybe hospitalized, and 3 months later, sterile. So I think there's a low perfusion rate there.
What is the phenotype of their sterility? Aside from the presentation that says I can't get someone pregnant.
So sterility means no sperm in the semen. And typically if you measured the signals to the testicle.
Oh, that's literally what it means. I'm sorry. Okay. So literally no sperm in semen. Yep. They stopped producing. Okay. Understood. Yep.
Right. So it's a primary problem. So the third thing we do, history physical, semen analysis is a third.
Yep.
Fourth would be hormones. And that's what we check in men too, because production of sperm is driven by the brain. So nothing happens to sperm being made without the brain telling it what to do. Similar with eggs and controlling. It's all a homeostatic mechanism with negative feedback. Classically, anabolic steroid users.
Yeah, which I want to talk about in detail. Can we go back to semen analysis? You're looking for, obviously, the count and the motility. What else do you look for?
So in the semen analysis, there's several features. I consider it sort of a poker hand. There's a volume, how much of the semen volume. There's a count, concentration of sperm, that's numbers per mil. And then there's motility, which is percent motion. You do a forward progression. So how good is the quality of motion? And typically some measure of shape called morphology. There's 3 liquid issues: liquefaction, agglutination, and viscosity. And then you look for other cells that aren't sperm. They're called round cells. And either they're going to be pus cells or immature germ cells that are ejaculated early.
And presumably you want to see fewer of those.
Right. There's a number like less than 1 million is normal.
Okay.
So if you ask me, well, how do I look at a semen analysis? That's a little different. I look at it as a poker hand with each card has a meaning, but they have a look. So if you said, what do you mean by that? So if the volume is low, it's one of 5 things. You're always going to find something. It's at the collection error. I call it first sample syndrome. Guy's not good at it. Right. You know, it's like, okay, I got to put it in the cup and I got to stop doing what I'm doing. And okay, so you do a second sample, and then there's low testosterone can cause it. There's an absent vas deferens, which means you have an absent seminal vesicle. There's ejaculatory duct obstruction.
And by the way, do you ever have that on one side and not the other? Mm-hmm. Or is that always, okay.
Mm-hmm. Yeah, no, it's very variable. It's segmental.
Yeah.
And so there's 5 real issues. So when I see a low volume semen as a surgeon and as I'm going to find something. So that's really good. Other than that, the semen analysis is I think I've been published as saying it's a blunt instrument for fertility. Unless it's 0, you can't really say much about their fertility because people conceive at all levels. And people
are there obviously you rattled off a whole bunch of parameters that you can access there, but are there certain null states that don't exist where everything is amazing, but this one thing is horrible? Like, do you see scenarios where everything is remarkable, perfect motility, but bad morphology, or perfect morphology?
Isolated example.
You do see isolated things.
Right. So one of them is I call it syndromic sperm shape problem. So you can have a perfectly normal semen analysis, count, motility, volume, progression, and the sperm look terrible. And so there are rare conditions, 1 in 5,000, where you might have globosospermia or two-tailed sperm or pinhead sperm. So if you look at shape, 4% should look normal, just terrible. We can have a whole discussion about why 4% of human sperm being normal is normal when 99% of animal species in the wild have normal-looking sperm. But it's all a construct. Right? It's all a construct of who someone decided what normal is. But in men who have large abnormal forms, like 4% is normal, if they're 1% normal, and you look at the abnormalities.
I'm sorry, I'm still confused on that point. Are you saying that you would consider it perfectly normal if only 4% of the sperm are morphologically perfect and 96% are not? And that means the 96% that are not could be pinhead, could be double tail,
amorphous or tapering it.
Going back to the original...
That's a mathematician. That's not a great number, is it?
No. And
if you look at marine species, 99.9 look perfect if in the wild.
Yeah. And presumably that's because their environment is so much more hostile. Like they're doing this all in the ocean.
I don't know, but it's amazing that we're that good with the sperm. But again, it's a construct. It's like ordering stars in the universe, Cassiopeia. Someone named Kruger said, this is what a normal sperm looks like. And But we know that two-tailed sperm have double.
Yeah, I mean, it might be the two-tail might not be the worst thing in the world if it's just extra rocket boosters. But what about the pinhead? What does the pinhead imply?
Pinhead means there's no nucleus. It's a tail with
so that's
a motor.
True problem.
It's like a little tiny head and moving along.
So if you rule out if you give somebody credit for their two tails, what does your normal go up to from 4%?
Oh, it depends, but maybe 20.
Okay, so there's still 80%
but
that would
most
fail.
of them are going to be amorphous. Head's a little rounder, head's a little narrower. Those are called stress patterns. And things like hot baths and varicoceles and smoking will do that, which isn't that bad.
All
so
right.
in the case of 1% normal, you got to look at the 99% because that's not the story. The story's in the other chunk, right?
Yep.
And if they're all looking the same, then it's syndromic, and then you have a problem.
I see. So the more homogeneous the failures are, the more likely that you have a clear etiology.
Yeah. And that's hard to fix. I mean, they'll fail with sex, they'll fail with inseminations, they'll fail with IVF.
They'll fail with IVF?
Yeah, they'll fail with IVF and ICSI. And sometimes with globosospermia, where they're called lollipop sperm, they just have a big round head with no acrosome. There's all nucleus and there's some of the components, they'll just bounce off an egg. They'll never work, right? They'll never work naturally. And to get them to work with IVF, you have to single sperm inject them into the egg and then shock the egg with calcium, do a calcium or piezoelectrics to get it to actually fertilize, because it also the sperm is important with fertilization. Not only has to bind, but the calcium channels are regulated by sperm. And what shuts the doors to polyspermy in an egg is calcium activation.
This is the reason why even if you launch 100 sperm at an egg, it's only one that can get in, because the first guy that breaches sets off the calcium channel that shuts the, I mean, the Star Wars space analogies here are they just phenomenal, right?
Million years.
Yeah.
A million years.
No more breaches in the hull.
So morphology can matter a lot, but it's very rare. So I'd say twice a year in my practice, I'll see this because everything's failing and everything looks normal. And they ask me what's going on. And I'll look at it really closely and say, you have this issue and there's not much we can do to treat it. Now we're going to try sperm sorting technologies, which are out new on the market, microfluidics and things like that. And I've been throwing that at them. Sometimes it works, sometimes it doesn't.
And is that something that we know the genetic underpinning of?
We're getting there. PLZ zeta deficiency is one of them, recently discovered, that runs the calcium channel, which tends to be associated with a certain look like globosospermia. So it's coming around.
Think about that for a second from an evolutionary perspective. That is the single least desirable genetic mutation you could acquire.
Yeah, unless you're not making sperm.
Yeah, but this is a dead end to the genome.
Right.
So does that mean it is only an acquired mutation, never inherited? I mean, it can't be inherited, presumably, unless it's homozygous, but even still.
Right. That's one of the 50 we throw off in each generation, 50 mutations.
God, that's just incredible.
I think what I would like to emphasize in this podcast is how fluid evolution actually is. And it's sperm-driven, and it's transgenerational. So if you ask me, what's the theme for today? I'd say sperm matter a lot. A lot more than we've given them credit for.
All right. So let's talk about, so basically just rounding out the semen analysis.
Physical semen analysis.
On the semen analysis, what if motility is the problem?
So I look at, in my poker analogy of the hand, if everything looks good but the motility is low, I think of short-term toxins. So not severe severity. So things like exposures, so medications. I would think about habits, pot, smoking, hot baths. I think about behaviors, lifestyle. I think about, so I look for an exposure in that individual. Basically picked up on the history, varicoceles and exposure, things like that. And if the count's down and the motility's down, I think of a more severe exposure. So there's royal flushes and there's 4 of a kind, there's 2 of a kind. So I would think of more of, my goal when I see that semen analysis and see that patient is to figure out if he's not normal, why?
And by the way, do you get that analysis the day he's in the clinic with you, or is that something you follow up on an appointment with?
Pretty much have it in my hand when I see them. either they give it to me or I get one. I want that there because that's when I look at them, I'd like to have that in front of me to say, what kind of poker hand are you playing?
And this is something that's standardized and automated through microfluidics? How is the assay actually done? So the guy ejaculates in a cup, takes it to a lab.
Oh, I mean, so it used to be done manually.
Okay.
And now it's done with hemocytometers and it's done with machines. Computer-assisted semen analysis does most of them in IVF groups.
So it's really standardized.
Oh, yeah.
Okay.
I like the bespoke suit. So I like the handmade semen. So when I have mine repeated, I usually have someone do it by hand because there's observations I like, which is, hey, you know what? 1% morphology, but all the others look like this. Mm-hmm. Those comments are incredibly valuable that you don't really get from a computer-assisted semen analysis.
Yeah.
But it's faster and you don't have a lot of human effort involved with the computer.
Are they using AI for this yet?
Yeah. I mean, some people are for sperm selection a little bit. But yeah, there's a lot of stuff to help out. And that will be really helpful for morphology to standardize it because one man named Kruger in South Africa correlated bad sperm shape with IVF outcomes and did not find that they were good when the sperm looked bad. That's where the 4% came from. But it's really hard to do that every time and do it well because it's so hard to do. Well, hoping AI and machine learning can help standardize the look because sperm, it's hard.
Yeah, given how good AI is at image recognition, this should be
tomorrow
a 1 foot putt. Yeah. Okay, you mentioned hormones. You were obviously alluding to LH and FSH. What else are you looking at? Testosterone,
right? So
estradiol?
To make normal amounts of sperm,
yep,
you need proper amounts of testosterone and FSH. Think of it as flowering a plant. You need the water and you need the sunlight. So if testosterone and FSH are key. To get normal amounts of testosterone, you're going to need LH, which drives it. Same in women. These are all named in women, in females. So that signaling is really important. So there are cases of genetic infertility, like Kallman syndrome, where men aren't making any sperm, but they're just not sending the signals down, and you can just give them the signals with injections.
Sorry, these men are not making FSH and LH.
No.
So they have virtually no testosterone.
Right.
Okay.
Nor sperm, but you can give them
synthetic
those signals. Yeah. Yep. So HCG, FSH injections, and they will be fertile.
Oh, so if you gave those guys, so is the problem in the pituitary, not the hypothalamus?
No, it's the olfactory node in the hypothalamus. So they don't smell either.
So could you give them Clomid and would they make?
No. Pituitary is not working. Yeah. It's the GnRH is
the GnRH is not
pituitary.
Okay. Got it. So, what about estradiol? Does it play a role?
Yeah. So estradiol is sort of a mild poison for male infertility. So everyone needs an estradiol level, female hormone levels. Testosterone gets converted to estradiol. Mm-hmm. So that's a byproduct of it, along with DHT. And then estradiol goes back to the brain and is a feedback. Yep. So if it's there, the brain knows how much testosterone it's making. So if there's too much estradiol, the brain senses, it's a negative feedback, senses, hey, there's too much of this, so let's make less testosterone. So it will lower your testosterone to have high estradiol. When estradiol is made, it gets metabolized differently than testosterone. It goes to the liver or to fat. And aromatases convert it to something else, or testosterone gets converted to female hormone and aromatases. So you can get high levels being obese mm-hmm. or having liver dysfunction, so alcoholic cirrhosis, hepatitis. It'll rev it up and it'll make a lot more estradiol level. And there's some medications that do it too. And that will act and lower your testosterone, which will lower sperm production because you're not watering the plant.
If you correct for testosterone, so in other words, if a guy has normal FSH, LH, and testosterone, is there an estradiol level by itself that is problematic?
Not usually.
Okay. So it's really only high estradiol in the context of suppressed testosterone.
Right. So that's when you would act on it.
Yep.
If you see that there's a low count and the testosterone's low, and you could say you need to lose 100 pounds, which is the key secret for everything, right? But you can also give aromatase inhibitors like weightlifters use to keep their levels down.
Okay. So those are 4 big pillars. Anything else besides the history, the exam, the analysis, and the hormones?
And then, so usually do 2 semen analyses 3 weeks apart or more to get a sense of things because it varies quite a bit. So a very important point is that the semen analysis, any feature of that semen analysis vary by 50 to 100%.
So never make a decision on one semen analysis.
It's really hard.
Yeah, especially if it's the first one, as you said, for all the potential.
So I could do a lot of consulting for the FDA, and they do they do medications in reproductive age men, and they're trying to show the semen analysis. They're going to the FDA, and they're saying, can you help us interpret this data for the FDA? I said, garbage in, garbage out. I mean, there's so much variability, you really can't say anything. It's really hard to get, so you have to do at least 2 samples, and it still varies quite a bit. There's inter-observer variability, who does the semen analysis. There's biological variability on what your system's like. So that's the big problem with studies, is that there's a lot of variation.
So what percentage of drugs that are going through an FDA approval process are having a semen analysis as part of the evaluation?
I don't think many.
So why is that?
Because usually the indications aren't reproductive age men or women. For some of them. If they do, they'll do animal models. They won't do human studies, do animal models. They'll do beagles, sort of mice and beagles. And if there's no fertility effects, they don't really look at semen analyses in those. They'll look at fertility effects in animals. If there's nothing there, then they'll probably not require human studies. If there's any suggestion of a problem in animal models, which is $1 million of work. So if you ask me why I patented the somatic gonadal stem cell, I want an in vitro test for human infertility that we could use instead of animal models. Save the animals, save $1 million, do an in vitro spermatogenesis model and see if there's an effect at all.
It just seems to me that in this day and age, with people reproducing at older and older ages, we shouldn't just assume that because we've developed a drug for blood pressure or diabetes, that it's not going to be used by people with fertility. I'll give you a silly example. Have GLP-1 agonists been tested for fertility? Okay, well,
because it's sort of an off-label use of a diabetic medication.
But it's no longer off-label.
I know.
It's an on-label use today.
But it looks like it might be helping with fertility.
But even if it was on-label, I mean, I'm just using that as one example of a drug that was initially approved when we thought, this is going to be for people who are not having kids. But the truth of it is, you're going to have lots of people that are trying to reproduce on many of these drugs.
Absolutely. And, there are 80,000 chemicals out there that are not been studied reproductively that are commonly used in industry. European commissions are a little better off. They've screened them and they've warned about them. But America,
Why is that?
I don't know. It's attention to detail. It's one of those things that just doesn't, I don't know.
Is it under the purview of the FDA?
Yeah.
Or the EPA? Probably
a combination, or maybe everyone's thinking it's the other person's job. I'm not sure. But they're untested and they're out there.
Well, why don't we just talk about some of those things then now? So this is, I'm sure, kind of a contentious topic, but as you know, there lots of discussion around microplastics, right? So I don't know how far we want to go down that rabbit hole. I recently did a podcast on this topic. I didn't really touch on fertility because I just didn't see any great evidence. I talked more about things where I thought there was a little bit more evidence. Obviously, with the microplastic story, there's quite a bit of smoke, but there's no real fire. My conclusion from the analysis was there is enough smoke that take steps where they are reasonable and reduce your exposure to these things. Everything from microplastics to PFAS chemicals to even
phthalates.
the phthalates and even the PM 2.5s. Like, there's no reason to expose yourself unnecessarily to this. If you can take relatively straightforward steps, eliminate 60 to 80% of them in your life, do it. Tell me what your impression is of the effect of any or all of the above on fertility.
So, although sperm are made constantly and are susceptible to that, we know the testicles are a pretty good place, and insulated from exposures. I also think there's a lot of smoke there, and it needs to be sorted out, but especially with the 60 to 80,000 chemicals that are being used that aren't really tested at all. I think the only way to know is to do stem cell in vitro testing as much as you can before you put it on at the investigational drug stage, not at the final stages for clinical trials, but early on do it. So you're screening them way in advance of getting into clinical trials and where the money gets big. But I think that there are windows of susceptibility in men, unlike maybe with women whose eggs are constantly exposed to toxins. Men have windows, and one of those windows is birth and early development, the first 12 weeks of life. So there's good data.
That early?
When all organ systems are developing, including testicles. Shauna Swan did this one with maternal beef consumption, estrogenized beef consumption. Their sons had lower sperm counts when they were 20 years later or something. So I think that's a window of susceptibility. I also think, puberty is a window of susceptibility when things turn on. So I think if exposures in those moments are probably going to matter the most to men. I don't know about other times.
And what is your advice to a guy when you're giving him counsel on everything he can do? We're going to talk about everything, but on this particular domain, if he says, hey, should I stop drinking Starbucks coffees in those plastic cups with the plastic lids? And should I get a reverse osmosis filter in the house? Like, where are you telling him to draw that line?
I'm not great at that because the stress level goes up so much. And I think the stress
counterbalances any amount of microplastics you save.
You double the stress in a man and his testosterone level will fall.
Yeah.
And then the sperm production falls for a whole different reason. So
my testosterone level when I left residency.
Oh, I bet.
So how old was I? 33. Should have had a pretty good.
Yeah.
Total T, 220 nanograms per deciliter.
Did you measure LH? It's probably low.
Yeah, I'm sure FSH and LH were totally low.
Right,
I don't remember what they
secondary
were. Free testosterone of like 3 to 4. Yeah. Well, the sleep deprivation, the stress.
So what does stress do? Stress is the sympathetic nervous system. It's fight or flight. You're running from a woolly mammoth. It doesn't know what you're running from. It doesn't know whether it's sleep or travel or financial or emotional. It's just the body. We are cats and dogs. We have the same binary nervous system. Either you're on or you're off. And when you're on, do you want testosterone? No, you want cortisol. You're running for your life. And do you want fertility when you're running for your life? In any species? No, you're trying to save your life. So cortisol goes on, testosterone is nowhere to be found, fertility is nowhere. You turn off all that stuff. Then when you outrun the woolly mammoth and you're behind a rock and you grab the berries and you catch a nap, boom, testosterone shoots up because it's rest and restore and you have to rebuild for the next run.
And how quickly do you think that occurs in humans?
How quickly?
Yeah, how quickly can that?
Days, easily.
We can reverse
chronic stress.
Yeah,
is it? It's not. We love acute stress. All species love acute stress, right? We love that starvation, intermittent fasting. It's really healthy, but not low-level chronic stress. Not connected to your computer, not your emails, not the workday that never ends. Terrible for us. And the best manifestation is erections, because the erections will fall if you're under stress too. Penis has a mind of its own, according to Da Vinci. I had a guy come in, he came in, he was like 25 in San Francisco, a startup guy, and he comes in and says, I got to see you. I said, why? He said, I lost my erection yesterday. He's 25. He said, first time? He said, yes. I said, all right, come on in. So he comes in and he's got his act together, looks good. And I said, what happened? He said, I just lost my erection. It's never happened to me before. I think something's wrong. I said, okay, tell me about yourself. He's just getting a round of funding. He's traveling 500,000 miles a year. He sleeps like 3 to 4 hours a night, if any, and he's constantly running. And I said, congratulations, welcome to the human race.
This is amazing. It's the first time. Yeah,
I know. And he's like, what are you talking about? I said, you're not impervious. Stress has its effects. So clearly, fertility, women who are sort of marathon runners. The great study was a moderate exercise men. I wrote a blog on this called "Can You Be Too Fit to Be Fertile?" Moderate exercise went to extreme exercise, measured as 2 hours a day of ViO2, 80% maximum capacity. So pretty heavy workouts for 12-week periods. So moderate to extreme, and then back to moderate. Sperm counts fell by 40% when moderate to extreme, and constant sperm testosterone fell by 50% and then went back up. So that's and there's also military studies of men under acute stress during hell weeks and training, where they were taking their testosterones and LHs and they were dropping by about 50% with severe stress. And that's okay for a day or two or a week. But when you're doing it chronically, we're not built for that, Peter. We're not built for chronic stress. That's a longevity issue.
Yeah. Let's talk about the use of anabolic steroids. So, or let's talk about it more broadly with sort of the 3 most commonly used approaches to testosterone replacement. So the way I see it is the 3 most common approaches are using either clomiphene or enclomiphene, using HCG, or using exogenous testosterone in one of its derivatives. Would you agree that those are kind of the big three? Okay. We'll just briefly highlight for everybody why each is a little bit different. Exogenous testosterone, you're just giving testosterone. The body senses it and immediately shuts down the hypothalamus.
All natural production.
Yep. Yeah. So LH and FSH will go to 0. Testosterone will be as high as you want it to be. There's no limit to how high it goes. I've had a couple of people on this podcast who have blown my mind with how much testosterone they've talked about taking. Not clear how that's possible, but nevertheless, they're doing it. Okay. HCG is synthetic luteinizing hormone.
LH.
So you give a person HCG, they will make testosterone. So it's endogenously produced, but they're making so much of it that they'll also suppress LH and FSH. So LH and FSH will come down, testosterone will go up. And then clomiphene or enclomiphene block the signal of estrogen at the level of the hypothalamus. Right. So the hypothalamus thinks,
doesn't see any.
Oh my gosh, we need more testosterone. Right. It ramps up FSH and LH production, which has the same effect as making more testosterone, but you'll now see high normal FSH and LH.
Right, and the two different classes are the LH and clomid versus testosterone. So unlike testosterone shutting off the natural production, the LH, the HCG, and the clomiphene and clomiphene will stimulate natural production. So you keep your testicular size, you maintain your fertility, whereas the others, you're going to shrivel up your testicles and not maintain your fertility. And you can't generate levels that you can with the exogenous testosterone with these. You'll never get to 3,000. You can't do that because it's tightly regulated.
So question one, if a guy is taking exogenous testosterone, and let's just say he's been on it now for a few months, is he able to create sperm?
Not 95% chance he's not.
Wow.
While he's on it.
Yep. Understood.
But can he create it once he stops?
And we'll definitely address that. But just to be clear, even a couple of months on exogenous testosterone in any form, injection, topical, oral, whatever, you basically have shut off the ability to make sperm because your testes themselves have shut down.
Right. No signals.
Yep.
Right?
Okay.
No gas to the engine. Now there's it's nuanced.
Okay.
So there are formulations that are topical that are less potent that way, less inhibitory than injectables. So there are variations in the spectrum of exogenous testosterone that will maintain some of your fertility. There are formulations.
At least the, I don't want to go so far as to call it the marketing material, but for lack of a better term, the marketing material is suggestive that the more frequently delivered variants, so for example, the intranasal variant, which is delivered 3 times a day, the oral variant delivered twice a day, have less of a negative impact because they're producing far lower surges than if you did a weekly injection. Is that what you're referring to?
Yeah, so they do more physiologic.
Yep.
They're in the normal range more. What gives you side effects from testosterone, including sterility, is
the spike.
Too much.
Yeah. So in your experience, has that borne out?
Yeah.
So you've seen men taking Natesto 3 times a day, doing a nasal.
Keeping their sperm counts.
Keeping their sperm counts. Mm-hmm. Okay. That's interesting to note. What about the oral testosterone, the twice a day?
Love it. So testosterone endecanoate, and it was not available in America for 50 years. It was available in Europe, and a couple of researchers at UCLA, a husband-wife team, beautiful, decided. What happened was we were worried when we took oral testosterone,
it would go to the liver,
right to the biliary system and go to the liver, cause liver cancer. So it was always verboten.
Even though there was no evidence this was happening in Europe for 50 years.
Yeah, it wasn't not much. It's FDA approved. The EEA approved it. So so this group came up with a way to get it metabolized through the lymphatics, so it can absorb through lymphatics and never hits the liver. Mm-hmm. And it's really good. I mean, there is a non-response rate of around 10%. So it's 10% of men. Some like gels too. 15% won't respond. There's groups that won't respond that well, but it is really good. And
Do
get
you
nice
prescribe
levels.
it?
Oh yeah.
You know, I'd be interested to hear your experience with it. We have prescribed it now to maybe a half a dozen patients. One of the silly challenges we have with it is we actually have no idea if they're therapeutic, because trying to get their blood drawn, to figure out when to draw their blood to actually see the level. For example, if a guy takes the drug at 8 o'clock in the morning and then at 1 o'clock in the afternoon, which is sort of what we're told is a great window to take it so that you get that
mid-dose,
right? If he does his blood draw at 7 o'clock the next morning, he's been 18 hours off drug. He has unmeasurable testosterone. He's going to show up at 200. He's going to look like I did 20 years ago. His LSH and FSH are still completely suppressed because that doesn't go away over 18 hours. But, I don't know how to interpret, like, what is he walking around at during the day, which is what I care about.
It's hard to know. I mean, usually you don't want to do it right away too. So you want to give them a couple weeks to stabilize hemostatically, right? But usually you can get pretty good levels because the half-life isn't that short.
They say it peaks in 5 hours. So I don't know what the half-life is. The half-life would be
like 12, more like 12. You wouldn't dose it at 100% decay, right?
Yep.
You would dose it at 50%.
Yep.
So he's probably not responding. We can check it at different times, but it's probably not much of a response.
And what are you dosing it at? It comes in 100 and 200.
So it depends. I usually go to the mid-dose 298 twice a day, and then you can double it or go half, whatever. I usually start out at not the lowest dose.
Yep.
And it depends what you're trying to solve too, the problem, right? If you want them, you're not going to get them to 800 or 1,000 very easily. You can get them, 400 to 600, 700 pretty well, but no side effects. I haven't seen anything. Maybe a couple dozen men, really well tolerated.
Interesting. So this is not something you used when you're trying to get a guy from 300 to 1,000.
You could, but probably not the first choice. Yeah.
And now you're taking 500 twice a day or something crazy like that.
Yeah, and twice a day is a big deal for men.
It is. Yeah.
And
what about the testo? How are you using?
You've never prescribed. No one tolerates that.
Yeah. What's the experience like? We've never used it.
Well, have a flu and try to get your testosterone level up. You can't do it. You have to spray it in your nostril, each nostril 3 times a day. And it's gooey and it's gel-like. And men within a week will call and say, can't do this.
Yeah, we've had more luck getting women to use this.
Maybe.
Yep. Yeah. Okay. So let's go back to the fertility of the other two. Yeah.
The two types of testosterone replacement or supplements. One is sort of, we'll call it the natural ones,
yep,
versus the exogenous ones, is the side effect profiles differ widely. It's very difficult to get polycythemic or thickening in your blood with the physiologic levels. It just doesn't happen very often. I don't think I've seen it once or twice, but if you take testosterone exogenously, you're at risk for polycythemia or blood thickening. So testosterone stimulates epipotin in the kidney, you make more blood. Athletes love it, but if you go on a long flight and you're dehydrated, you're going to throw a clot. And people look at it for longevity and it's like, be careful.
Yeah.
Because I've seen 70-year-old men want longevity and taking this stuff and then they have a clot and they have a stroke and now they're 71 and they're not, the whole.
And do you find that the clot risk is proportional to hemoglobin hematocrit?
Absolutely.
Simply? And at what level are you saying it's time to train?
I mean, the studies aren't broad, but Ramasamy just did another paper on it, and the most significant event occurring with testosterone replacement or supplementation is polycythemia and events. And so the high level for hemoglobin 17, hematocrit 50. You start seeing events happen about 18 and definitely at 19.
Yeah. And one of the things we do with patients who are injecting testosterone cypionate is, and we have some patients who love doing this. I think it would drive me nuts if I were trying to do this. They inject it every day. So they'll do 10 to 15 milligrams every single day. And it actually produces the same effect, which is they don't have the polycythemia.
Right, because they don't hit the peaks.
They never hit these crazy peaks. 10 years ago, everyone I saw that was prescribing this was prescribing, the standard was 200 milligrams every 2 weeks, right, which was crazy.
Highest risk, yeah.
So what is your typical injection schedule? Once a week or twice a week?
And I think twice a week you can have the dose, right? If
yep,
you do it. So that is a little safer, but then it becomes the intensity and just a bit, I can't do it like that or whatever. I want a pellet instead.
Yep.
So
do you put pellets in?
Oh, yeah, I do them all.
Yep. And do you find that the pellet also, I don't know the kinetics of it, but I would imagine you're pretty supraphysiologic for a month or so, maybe?
Yep. So pellets are like the long-term contraceptives for women.
Yep.
In the arm, they put it subcutaneously. We put it in the butt. And it's a couple-minute procedure in the office, and it gives you don't have to worry about anything. There's no compliance issues. We don't have a lot of side effects or consequences from it. It's done with a trocar and a thick needle. And you get, pretty quickly, within a couple of days, you'll get a level, and then it'll slowly decay, pretty much half of it by 3 months or so, and then the rest by 4 to 6. It's supposed to be a 6-month physiologic level, but normally it's 4.
Yeah.
4 or 5. And men feel great for a while, and they can feel it because it's slow, but it is even, and you do have this risk of polycythemia and things like that. But there's a 3-month period risk, and then usually there's when you're in the normal range, it kind of goes away. So I don't see a lot of consequences with that if it's 6 months. I really don't.
So let's go back to the Clomid HCG route. What is the extent to which fertility is preserved when a man is on one of those agents?
So Clomid we give it for fertility all the time. So I'd say very good.
Yep.
It might even improve it. HCG depends on the dose. So like you said, high doses suppresses. So to be normally fertile, you want LH and FSH going to the testicle. You want the water and the sunlight. You want the testosterone. If you've got the testosterone, but your FSH if you don't have any sunlight, you're not going to bloom. So I usually add Clomid to HCG if the dose is above 1,500 units 3 times a week, because that's going to start suppressing the FSH, and Clomid will keep it going, and then your fertility is preserved.
1,500 3 times a week of HCG is a whopping dose. You're saying beneath that, you typically don't have issues with FSH and LH suppression?
Right. Well, LH you will, because it's LH.
Yep.
But not FSH. No. Maybe 1,000 to 1,500 you start seeing it.
Okay.
So that's why you protect the fertility.
Yep. And you will just give, and what dose of Clomid will you give on top of that regimen?
Depends. I mean, usually half a pill a day, pill every other day.
Half a 25, half a 50?
50 milligram pills, yes, usually half.
You'd give 25 every day?
Huh?
These are staggering doses. How high are these guys' testosterone getting?
The testosterone is driven mainly by the HCG. So I shoot for the normal range of 500 to 1,000. I don't.
Yeah, that's
I'm
amazing.
not an anabolic guy. I'll note it today.
Yeah. No, it's interesting. I mean, we don't like clomiphene at all, just because. Well, there are a whole bunch of reasons, but they have to do with kind of lipid stuff. But even when we would use it, we would probably use 50 3 times a week. So that's about the same, right? 25 a day. But for most guys, that would be sufficient alone, even without HCG. But you're saying, okay.
Well, it's the HCGs that's driving the T. We're just trying to protect it. If you say, well, what do you give on isolation as monotherapy?
Yeah, what would you give for Clomid monotherapy?
12 and a half to 25. Typically, depending on how sensitive the system is.
And do you prefer Clomid and clomiphene?
So it's very interesting. Clomiphene's really good. It's an interesting FDA story. So Clomid is not approved for men, and clomiphene isn't either. Clomid's approved for women, and clomiphene's not approved for either. And clomiphene's compounded. Clomid is available for 50 years, so a lot more data. And one's a cis isomer and one's a trans isomer, so they're different, and the estrogenic effects are slightly different. So I have enormous experience. I have 560 men on Clomid, and I have fewer in clomiphene. But it was developed for older men to preserve their testosterone levels as they age, because the signaling tends to get weaker. The pituitary tends to get lazier, and this is to keep your testosterone levels up more physiologically than taking testosterone. So it went through some very good randomized trials that were published.
This was clomiphene or N-clomiphene?
Citrate, okay. Clomiphene. And they were done by reputable people in the field and published, and then it went to the FDA for approval for secondary hypogonadism, sort of age-related changes, not primary testicular failure, in age-related androgen deficiency of the aging male, or ADAM, right? FDA sat on it for a couple of years and said, nope.
Why?
Good question. So it's published, they're good trials, it's safe, it's as good as Clomid, and they didn't approve it. And I think it's hard to know, but I think the reason was that there's such an uproar about testosterone in America right now, and the FDA doesn't like what's happening. what happened is you can advertise your drug to the consumer now, right? So all the biological response modifiers for psoriasis, all those drugs go on and they give you 5 seconds on the benefits and the lesions go away, and then 25 seconds on side effects, right? So you can do that. If you do that with testosterone, what you hear is, do you falling asleep after dinner? Are you not as athletic as you used to be? Are your erections not as good as they used to be? There's 10 questions in the Adam questionnaire, and every guy's
going to be like, yeah.
Everyone who ages has those issues, right? So it's a no-brainer. If they go on TV, they're going to want this stuff. So the cat's out of the bag, and they're stuck. And so now when any testosterone trial comes back, they're going to point out, the FDA makes sure that we point out the dangers of testosterone replacement, okay? So this is part of that energy, which is, we don't want another testosterone.
So I think there's another reason, Paul, and it's everything you just said, but HCG and testosterone are Schedule 4.
True.
Which means you cannot prescribe them through like these testosterone clinics that don't even see patients and, are literally just not being doctors, right? They're just sort of giving it to anybody who shows up and pays. It's a coin-operated testosterone dispensary. But Clomid, and I assume by extension, and clomiphene are not scheduled.
Right. Which
means you can coin-operate those. And my guess is that's probably why the FDA is saying what it's saying is it's already bad enough that the Clomid cat is out of the bag, but given that we don't want to put another one of these unscheduled drugs out there in the land of sort of shady medicine.
And the indications are pretty clear, and they're really safe. They're really safe drugs. And I like my effect is someone comes in who's young, who maybe wants kids, hasn't had them, and they have a low testosterone of 220. You measure their LH, which no one does. It's low. Secondary hypogonadism. So it's not a testicle failing.
Right.
It's a signaling issue. And that's probably stress. So I said, get rid of your stress. And they say, how do I do that? It's like, well, okay. So exercise, acupuncture, massage, or yoga. I mean, for men, I say physical activity is the best thing for sex. So as an aside, during COVID, I had 2 groups of men. They said, what do I do? My life's a mess. Everyone's life's a mess. So half of them had drinks at 5 o'clock and started drinking a lot. And the other half went out for runs or got a Peloton, which most of the country did. That's a great story, the Peloton story. And then about 6 months later, these guys realized it's not working. And they started shifting over to exercise. So I was very proud of them. These guys I was really happy with, like, nice. Because that's the best way to handle stress is when you have no control over things, go for a run, go for a walk, get out there. Yep. It's just so good for you. It's just decompressing, get your mind off something, anything, surfing, whatever. So they don't do that. So I said, well, let's do this. I think what's happening is this. I think it's just stress. Maybe try traveling less or whatever. And then I'll give them Clomid. I'll say, let's try this for 3 to 6 months. And let's see how you feel. Sometimes it's sexual health issues. Erections aren't typically that dependent on testosterone. Typically it's other things. And so if that's the issue, and I'll say, I'll give you the benefit of the doubt. Maybe you were higher before, and we don't know that, but let's do something pretty safe and easy, and I'll double your testosterone or triple it. Let's see how you do. And then I'll check in with them at 3 and 6 months. How are you feeling? I feel great. Or, hey, it's not working. I feel the same. I say, well, it's not testosterone related. Whatever the symptom is you're having, you wouldn't have it with a testosterone. We know levels of testosterone above which you should not have symptoms.
Right.
We know libido, we know erections, we know fertility, things like that.
And what are the approximate levels for each of those?
I don't know. Erections, I would say the best study is about 290.
Yeah. So most guys that are having difficulty with erections are above 290. There's some other issue.
Usually.
Yep.
But you have to prove it to them. And I'm fine with that as long as it's safe, right?
Yep.
And that, you're convincing them.
And then what about libido?
Libido, I'd say 350 is sort of a range. It's pretty sensitive, and it's harder to call because libido is driven by so many different things.
And fertility?
Fertility, I'd say 300 is a good one. You start seeing issues
with
below.
How much FSH and LH?
I don't know.
Okay. And then obviously the other thresholds would be anabolic capacity, like muscle mass and things of that nature. And mood tends to be a lot more variable in my mind.
Absolutely. I think there's myths around testosterone, and those are some of them. But testosterone is a, sort of a Morgan Taylor and equilibrium story where if you're low, you have symptoms and you're low, those symptoms will get better when you go up. But then there's a point where it flattens out and there's no increase or improvement in symptoms. Sexual health symptoms are classically ascribed to that. There also is a linear relationship between testosterone, and that would be blood and muscle.
Yep.
So more is better for making blood, doping, and also blood doping, and also for muscle. Absolutely linear.
Yeah. And I'll tell you why I find that interesting, Paul, and I only learned that really in talking to bodybuilders who were taking 500 to 2,500 milligrams of testosterone a week. Because my initial reaction to that was, you've already saturated the androgen receptor probably 5 logs. I mean, not 5 logs, but at least 1 or 2 logs earlier. Why? How would you still get but they convinced me, no. There's a real difference between 500 and 1,000 and 2,500 in terms of muscle mass, which it sounds like you agree with. And I don't understand the physiology of how that's possible. How many androgen receptors would you need to? You'd have to upregulate them when in fact you'd be downregulating them.
So I'm not sure, but it's not it. So the effect is indirect. The effect of testosterone on muscle mass is indirect. It's not that you're going to do it and create mass. You don't just create mass. What it allows you to do is recover from injury, right? So if you push the system and you need 2 days to recover, you can go to 1 day, you can push it again harder. So that's what testosterone does in the primitive world, right? If you're a caveman.
There's even studies that show, by the way, that high enough doses of testosterone will increase muscle protein synthesis absent the stimulus. Absent the lifting stimulus.
So it's the potential to recover that is improved. And I'm not sure that's receptor driven at all. Yeah. Like it might be several pathways going on that are logarithmically better, but it allows you to push the system and go back and then push it again. And that's how you build muscle.
All right. So now let's talk about the guy who comes to see you. He's been on exogenous testosterone for 3 years. So he was given poor advice 3 years ago. He went to some shady, back alley website. And they just, he was 27 years old at the time. He just wanted to, this is tragically a very common story, by the way, right? So this guy's been on 200 milligrams of testosterone a week for the past 3 years. He's now 30 years old. He's met the love of his life. And lo and behold, they can't seem to get pregnant. So he's in your office. During the history, you find out pretty quickly he's been on 200 milligrams of testosterone for 3 years. Tell me what his sperm analysis looks like. Do you see presumably there are no sperm?
I would bet 95% confidence that he would have no sperm in his semen.
Okay.
At 200 milligrams a week.
So what are you telling him now? How are you going to solve this problem?
So it's funny because a lot of guys come in, they look good. And when I examine, I'll say, are you taking anything? Because they never put it on their medications, right? You never write it out on the history. You always have to get it out of it. And if
they're super jacked, but then they have shriveled testes.
Yeah, and they're zero. And they're wondering, right? So I will look them in the eye and say, are you taking testosterone? And I'll look them in the eye until they answer. And if they look down, and they don't say anything, I know they're on it. If they look me in the eye and say no, then I know they're not. But they'll always look away because it's this verboten thing. So
this is the same, by the way, as I'm sure you experienced as a resident in the ER. The people that come in with foreign rectal bodies and abdominal pain, that's the one thing they omit from their history, right? It's like they tell you, this is the last time I ate, this is this, but then you get the x-ray back and there's like a candlestick in their colon. And then you say, what about this?
Your Hopkins.
What about this? What about this candlestick? And they're like, oh, I totally forgot to mention that. Yes.
It was lit when I went in.
Yeah.
So typically I'll say, so my theories about this is why is he taking it? So if he's taking it for anabolics, then he probably has a pretty good idea. And I want to give you a little research we're doing on the lifespan of anabolic steroid users. So remind me at the end of the story to give you a little brief about what I know about that. But so how he takes it matters. So if he's been in constant use, injectables, that's the most suppressive of fertility. And if you turn a gland like a testicle off long enough, it's off. So I gave a lecture to the Endocrine Society on recovering men from hypogonadism in young men. And I asked them a question at the end because my whole procedure comes from steroid users. I take notes when the anabolic guys come see me because they're really smart and they know a lot about reactions, biology.
Yeah, it's incredible.
But not everything.
Yeah,
but it's a science. Some of them are PhDs. I took notes for years and it came up my approach along with what I know, right? So it's very much in concert with them. So everything I say is sort of built on a large experience, and it's called Getting Off the Juice, the blog. And I have people read that blog, do it, and say get about 80% of the way, and then call me and say, I need help here. Now I'm here.
Yeah, we'll link to this in the show notes.
It's pretty cool. Getting Off the Juice. And there's a PowerPoint in it. So the recovery is usually possible in young men, but it depends on how much they took, how long they took it, and how they took it. If they do it like a cycling effort, that's the best, right? So if you cycle steroids, you recover the pituitary, you get back to normal, and then you hit it again. That's actually quite smart. Constant use is not. Constant use for longevity or whatever is not a good idea for fertility. So that's going to be much more suppressive. Injections are worse than orals or any gels.
Mm-hmm.
So the next thing is how long. So I asked the Endocrine Society, since I answered all their questions, I said, I have a question for you. Can you turn a testicle off like in a thyroid or an adrenal gland? If you suppress it enough, can you turn it off for good? And they said, yeah, that's a board question of ours. We can do that. And I said, because we believe it's always reversible in the field of infertility in men. And so that got me a little worried. And so now I kind of worry about 5 to 10 years of use. After 5 or 10 years of use, you may not get it back, either the ability to make sperm or the ability to make testosterone native.
So we typically tell men in our practice, don't, 2 years would be the absolute ceiling. Maybe, are we too conservative?
Maybe.
Okay.
Depends on dosing and everything, right? Yep. If they're taking 250 a week.
No, I mean, in our practice, it would be 50 twice a week. Yeah. Okay.
I did published a study when I was a fellow in Houston of a guy who took it for 25 years. And we drove at him with gonadotropins, that's HCG and FSH, and we didn't get anything, but he had we got a low number of sperm back. And I just had a guy from Louisiana come in, 25 years of chronic use. I did a mapping procedure to find sperm in his testicle, and he's going to be having a kid, but he made a couple of sperm. And that was a record for me.
But you pump him full of HCG and synthetic FSH.
And get nothing, and then you have to look in the testicle because production can be low enough to be there, but not coming out.
But this is the rescue protocol. It's LHFSH.
Basically, there's 3 ways to do it. One is never stop the testosterone suddenly.
Interesting.
Because men will hit the doldrums and go boom, and they'll flop over like they have the flu. They'll feel like shit, and they'll get right back on it. They'll feel terrible because they have nothing going on. If you take the testosterone away, their system's turned off. They're not making their own. It takes time to get the system to reactivate. So that's the hardest. So I always taper testosterone over
what period of time?
6 weeks. Typically, you have the dose for 2, and then off for 2, and then you measure. And that's like getting out of the white water into the green wall a little bit. So that's a little smoother. So taper. And then I offer them 2 options. One option is taper alone, taper with Clomid or enclomiphene, which is a little quicker, getting the pituitary to turn back on. So that will soften the blow of the feeling of feeling completely fatigued, or more aggressively HCG and Clomid.
Okay. That's interesting. I guess there's no
at about 6 weeks,
and there's no. So it's interesting. If you give Clomid, the pituitary will make FSH and LH.
Yeah,
that doesn't shut off.
It takes a while.
Well, that's a way more cost-effective approach than giving because synthetic FSH is pricey.
Yes, a couple $1,000 a month.
Yeah.
In America, yeah.
So is there any reason to do that over the Clomid approach, or is it just that it's faster?
I think you might gain a couple of weeks of time.
Okay. So for most people, that's not a price worth paying.
With that taper over a month or 2, I usually check their T levels at around 2 weeks off of the last testosterone, and that's the lowest they'll be. And if they're in a good range there, you can use that as a predictor of their response. If
what would be good?
If they're in a normal range.
Oh, really? Okay. So we want within a couple months to see them back to 600.
300 would be okay.
Okay.
To make sperm.
Okay. All right.
But then to get them to where they want to be depends on their symptoms and what they're happy with. I'm not sure. You won't know until you wait longer to see how high you can get them. That's the lowest they'll be, but they'll be off of testosterone. And if they go along that taper and they're not tolerating, I try to tell them, don't go back. Just stay there because time will help you.
Yeah.
And you're not going to feel maybe that great, but try to do this because if you don't, if you go back, then we have to start over. But if you can just maintain it for a while, you'll feel better. And some of them dip a little bit, but remarkably, most men do really well with that taper.
Now, before I want to get onto some of the other topics here, but just to close the loop on this, do you ever advocate kind of crazy ideas for guys that are using testosterone to use lower doses and then combine it with HCG, just as we were talking about the Clomid plus HCG approach?
All the time.
Okay. So not an unreasonable approach to combine Clomid with testosterone at low doses to preserve testicular function.
I operated on a man, a testicular sperm retrieval on a man who was asospermic for genetic issues, and he was on testosterone for 10 years. And because he needed it, his testicles were failing. And I said, you're not going to make sperm on this. So we put him on HCG, which didn't do anything for him, felt terrible, and did that for a year. And he said, I can't do this anymore. I said, okay, or maybe it was 6 months. And I said, I need a little more time for you to be off testosterone. But since you've been on HCG for 6 months.
And what dose did you have him on?
Like 3,000, three times a week.
3,000 three times a week? Yeah. That's a whole vial a week.
Yeah. Yeah.
Wow.
Yeah. Then I said, okay, let's add in a low-dose T-gel, testosterone gel, to get your testosterone up, and we're going to lower the HCG to 500 3 times a week, twice a week. And I did a sperm retrieval yesterday. Boom. Plenty of sperm.
How old was he?
35.
Interesting.
So you can maintain whatever's going on the testicle with HCG and take any testosterone you want. That's an
important lesson. Yeah.
It was, here's the catch though. The caveat is it was done in, I think, Finnish bodybuilders. They were doing a cycle of steroids, huge amounts. They took low-dose HCG 500 twice a week. And John Amory has worked out, in Washington has worked out all the exact doses, but 250 to 500 twice a week is a good dose for that. It keeps your intratesticular testosterone high, keeps your sperm production going. And they went on both concurrently for 12 weeks, and their sperm counts were normal the whole time at any dose of T. Now, what happened after that is people start saying you can preserve your fertility on testosterone replacement, which is possible.
They missed half the story.
But it was only 12 weeks. And if you're doing it for 3 years and you miss your dose of HCG, boom, you're done. You're cooked. You're going to go to zero. So unopposed testosterone without it, so you have to be 95% compliant.
And do you think that there's a difference between HCG and Clomid in that effect as the adjunct?
The Clomid doesn't work even near.
HCG is the one.
Yeah.
Okay.
Clomid doesn't improve intertesticular testosterone levels like HCG does. It's ineffective.
Got it.
It will potentially make you more recoverable. If you do it 80%, you'll be zero, even though you thought you might have a sperm count. But your recovery will be faster because it's done something. But the only way to maintain your current fertility is you have to be 100% compliant with dual therapy. You can't go on monotherapy with testosterone.
Outside of fertility, given the popularity of testosterone replacement therapy today, is there another advantage to just doing dual therapy? Obviously, for fertility, we wouldn't be talking about it, but can you think of any other reason why it might be advantageous if a guy can deal with the hassle and the cost?
Yes, depends on the indication, though. I mean, if
everything but fertility, like any other health benefit?
Well, I think muscle mass. So with aging, it's a great one. I mean, it used to be like growth hormone with age wasting syndrome, things like that. I mean, muscle mass is a key for men, right, as they age.
But I'm saying, as opposed to just being on testosterone injectable, to do the dual therapy versus just monotherapy.
Oh, you mean if you're going to do some kind of therapy?
Yes, if you've committed to doing therapy.
No, I think the only reason would be if you want testicles to be big.
Got it. Okay, so just volume.
I just created a new procedure to make testicles larger naturally by putting a fat injection in the hydrocele space in men on testosterone because they don't like their small testicles.
So it's the equivalent of the Brazilian butt procedure for the testes.
Yeah, so it's all natural and there's no prosthetics and that you can't tell, and it makes them nice and big, and it's called testis fat grafting, and it's fabulous. But that's That's what
Medicare
you have to
improved?
do. No.
Yeah.
Creatively approved.
All right. So let's shift gears and talk about other modifiable factors. Let's talk about heat. We've talked about it a little bit.
So for fertility.
Yes, for fertility. So tell me about the impact of cold plunging and sauna and hot tubbing on fertility for men.
Okay, so the testis is outside the body.
Yep.
It's 3 degrees cooler than the rest of the body, so 95 versus 98 degrees Fahrenheit. And then there's a reason for that, unknown. We had that conversation, don't really know why, but it may be that it's an immunologic sanctuary and that's the only way to do it, and that God or Darwin could figure out. But if you heat up the testicle, it's also close to the skin. So it's a radiator. So when the heat comes down the arterial blood, it has to cool. So it raises and lowers. And there was an article in the Journal of Irreproducible Results about 20 years ago. A man went to Big Sur and wore nothing. And he measured ambient temperature, and then he marked on his leg with a marker where his scrotum hung, how low it hung. And he could tell the ambient temperature by how high or low his scrotum hung. He became a thermometer. So it does go up and down. Is that Peter or Tia laughing? Journal of Irreproducible Results.
Oh, God.
Really cool. But it showed that it's very temperature sensitive, right? And it goes up and down to regulate it closer to the body when you want it warmer, et cetera. You go into a cold shower or a plunge, where are your testicles? They're way up there.
Right? In my abdomen, yeah.
And that's all the cremasteric muscle, and it's all temperature driven. So it spends all of its time regulating its temperature to stay at 95. Now, saunas, baths, hot tubs, jacuzzis, steam rooms change that, right? The worst one of those is anything underwater, submerging underwater, because you're 1 centimeter away, you're a liquid, it's a liquid, you're going to turn that temperature, maybe not the inner part of your body, but little kids going into hot tubs, right? They overheat. So testicles will certainly overheat.
So you get into a 105 degree hot tub, which is a very typical temperature for a hot tub is 105 to 110. You're saying within a relatively short period of time, your testes will assume that temperature.
Absolutely.
Yeah.
You're 70% liquid.
Yeah.
And this is right at the surface. So I did a study, published it in the Brazilian Journal of Urology. I published 200 studies. This was the hardest one to get in. Everyone said, we know that it affects fertility, so we're not going to publish it. So I went to the Brazilian Journal of Urology. It then went to the New York Times as a press release. That's how popular it was. It's probably my most cited paper ever, and it's certainly not my best. It's very interesting. I took infertile men with low sperm counts and stopped the tubs. They were in hot baths because I used the word jacuzzi. Jacuzzi called me up and said, stop. Don't use that word. So I don't use that word. So hot baths or tubs. And I told them out, and they went up 300%. Their semen quality went up 300%, total mole count in 3 months or 4 months, and 600% at 6 months. So, you have to give it some time. And that's that curve, right? The recovery curve. And we didn't look at fertility. We just looked at that recovery. And some of them were 0 and went up to close to normal.
What was the age range of these men?
35.
So fertile men.
Yep. And trying to conceive.
Yeah. These are men who are not able to conceive. You're making the diagnosis. I think it's your hot tub. Let's get you out of there. And they have a 6-fold increase in sperm count.
Total sperm count, meaning count motility, mainly driven by motility.
Interesting. So it's motility that the price.
The biggest one, but also count might have doubled. Fertility may have gone up 3-fold kind of thing. So 6-fold increase overall. Then after that, I calculated a lethal dose of tubbing. So what's the lethal dose?
Yeah, what's the LD50? Yeah.
So lethal dose to me means you're 0.
Yep.
You do it enough, you have no sperm. And it came out to be 20 minutes of a hot bath or tub, 20 minutes on 104 degrees, 3 times a week would probably make you 0.
Wow. There have to be a lot of guys out there who are spending at least 3 times 20-minute sessions in a hot tub that's at least 104 degrees a week. And
the highest group, the largest group of people in tubs in Northern California, we did the study, were environmental lawyers. I don't know. It's like, is your job that stressful? It's like, yeah, it is. I mean, it probably is in California.
All right.
So then I think there's been studies. That was the only study ever done prior to that was a PhD thesis at Vassar College, where someone had a guy dip their testicles into a bucket for 20 minutes at really hot and looked at their sperm counts or their fertility, and that was the only, and I couldn't even find it wasn't published. I had to go, to figure out this thesis thing. But that's how little was written about it. And they gave me so much flack for publishing this. It was really funny. And the New York Times had an article said, it drew a condom, and it drew birth control pills, and it drew a guy in a tub. It's like pick your contraceptive. So it's huge. And I asked it, I'd say 10% of my population's in it. And then the next question is, what about saunas? So saunas is not underwater. It's not submersion. But saunas are, you're in a hot room. It's going to affect it. And I would say the effect is 1/4 to 1/3 as profound as a hot bath or submersion.
So my friend was absolutely right to have those ice packs on his scrotum.
He's reading.
Yeah.
He's listening
he's a smart guy.
The Turek protocol.
Yeah.
And then I would say steam rooms, showers are probably fine.
Yep.
Because you're in an ambient temperature is normal. And I think steam rooms are probably between saunas and, it depends how much time you spend, but it's probably not normal, but not a hot bath. Hot baths are terrible.
Okay. And then what about the cold?
I don't worry about cold. I remember Surfer Magazine called me and said, I'm a Northern California surfer, right? Now LA surfer. And they said, the editor of Surfer Magazine called me and said, are surfers infertile? I said, is that water bad for them? Because California water is 60 degrees. Yeah. And I said, no, I've never met an infertile surfer. So I don't think it's bad at all.
All right. So the cold is okay.
Especially plunge where you're talking seconds.
Yeah. Yeah.
Your testicles are going to go up and you're going to be able to maintain that heat. I think if you did it all the time, it would probably be bad. Yeah. Because enzymes work in the testicle at that one temperature. They work optimally.
Okay. Let's talk about exercise. You mentioned one example of exercise that can be problematic, which was, I believe you said, in a study where men were ramped up to 2 hours a day of exercise that was above 80% of VO2 max, correct, which is pretty strenuous.
That's right.
That was enough to put a dent in their fertility. Tell me about riding a bicycle.
I'm a biker. I have old vintage bikes that I used to race in Connecticut, and I had them rehabbed, and they're all Italian, and they're all steel, and they weigh a ton. And the seats are from Britain, and they've got 10,000 miles on them, and they weigh 4 pounds as much as
just like a Brooks saddle. These Brooks leather saddles.
Yep. And all worked out. And it's like that saddle nowadays is about half the weight of a carbon bike, right? Yeah. So, but I love it. And I was thinking of maybe going senior league and doing this gorgeous steel frames and trying to keep up with those guys, because it's not about the bike, really. It's like when you get golf clubs, and I got a $150 set of golf clubs. I'm going to be as bad a golfer with $1,000 clubs as $100. So it's really about the biker. But there are some differences, in terms of the momentum and the wheel force and all that. But I love my old steel bikes. So, and they see this. It's like I hang it in my office when I come to work in the morning, and I bike in San Francisco, and then I have the seat. And it's like that's a bad saddle. And so the issue really is it got started that biking was bad for reproductive health with a Spanish competitive cycling study. So competitive Spanish cyclists, Tour de France caliber cyclists, their sperm counts were examined.
But what was the control group? Did they have a control group of runners?
I don't remember. I don't think so.
So in other words, it could have been the exercise it could have been the intensity of their exercise.
Not a good group to study.
Yeah.
So their sperm counts were low, their morphologies were off, and they're extreme athletes. So we know that, and we know maybe they were on drugs, maybe they're, it's a big industry. They're super fit. They're certainly exercising 2 hours, right? And so they said, look at these guys who are really healthy and look at their sperm counts, but this other data didn't come out. So I did a blog called Cycling into Childlessness. And I looked at a more comparable study, which was British commuting cyclists. So everyday people bicycling to work in Britain on different saddles and I looked at their fertility, and their fertility was far better than the average Brit.
So even if they were taking some hit off the bike, it was probably more than compensated for by their healthy lifestyle, which included probably riding the bike. But obviously there's a healthy user bias, because anybody who's riding their bike to work is probably consuming less Guinness, fewer fish and chips, smoking less. In other words, riding a bike is a proxy for being healthy, but in spite of that, it didn't offset that health risk, at least unless we found people who were equally healthy who didn't ride a bike. I don't
remember what they controlled for, but I think they did a lot of the socioeconomics.
Okay.
And it was just, it may just been activity, but the bicycle.
So is this a myth?
Yes. Now, if you said, am I worried about bicyclists? Yes. So I worry about sexual health. I worry about the pudendal nerve, and I worry about seat anatomy. So the best seat for a bicyclist. So if you're biking a lot, that's good. If you're biking and you're getting pelvic numbness, that's bad. So you need to get a better seat. The best seat was studied by the NACH, I forgot. It was Dr. Schrader at the NIH. The best seat is, the saddles that are shaped like this are bad for your sit bones because they come into the middle where the arteries and nerves are to the penis. So it's an erection issue. Those aren't good saddles. The saddles with the two little tongs that hold your iliac crest bones with no nose. Perfect, right? So it's pressure, where the pressure is, outside facing, leaning in. So we gave those to police in Washington, the bicycling police down in like National Cathedral area in the parks. And they all gave the seats back a week later, said, I'm not doing this. And he said, what's going on? He said, we don't know where the seat is. We go sit down and it lands somewhere. You have to have the nose for bicyclists because they use it to guide when they sit down, they use it to guide where they sit. So the best saddle is flat or gel in the back, cut out in the middle, and some kind of lean in like this. So cut out saddles, and then you should get your bones fit. So you can do this online. You can ask them to send you a pressure pad, and you sit on it, and then you send it back, and they measure the distance. And there's only a couple of different saddles, maybe 12 widths that you could do, and you get it done. Or like me, you use a saddle used for 30 years, and it's perfect, but it weighs 4 pounds.
But that's because it's a leather saddle and it fit to you.
Yeah.
No, they're iconic. Okay, let's talk about alcohol specifically, and let's talk about any other recreational drugs.
Toxins. Yeah. So fertility-wise, I'd say the government wants men. I'm going to talk about men to less than 2 glasses of alcohol a day is okay. They consider 4 binging. Now, alcohol is a small molecule, goes right into the brain, goes right into the testicle. It's definitely a poison, right? It goes everywhere. The testicle doesn't limit it. So I worry about it a lot. The effects I see are direct when it's abused. So I would say you see morphology, motility, and count issues. So that's a direct effect as a direct toxin because it's one of the few things that gets into the testicle. Second would be a hormonal effect. So alcohol use tends to cause the liver to rev up, tends to cause more estrogenization. So you tend to get low testosterone from that. So it's a hormonal effect and a direct effect.
Any evidence that it's having an epigenetic effect?
God, probably. No, I don't know about evidence, but I'm sure it does.
Let's talk about common recreational drugs. Let's start with marijuana.
That's the worst player for me. So THC, same thing, count motility, morphology. So it probably has an effect. We know it has an effect on fragmentation, which is a quality measure of sperm, not only the way it looks descriptively, but quality, and also probably an epigenetic effect. Some of the early studies on sperm epigenetics showed alterations with nicotine and with pot. What I don't like about pot is depending how you ingest it and however you ingest it, you get a peak, you feel it, it goes away, you feel it's out of your system like nicotine.
But it
sits in your fats for a month or 3 weeks, and it's a depot effect, and it keeps coming back. So you get a low-level toxicity, which I don't like at all. So I am not a fan of pot. The other thing that really concerns me about pot in reproductive age men is I wrote a couple blogs on this. It's called The Weed Worries. And there's some compelling evidence from epidemiology in 2 studies, 10 years apart, validating each other, that chronic pot use is associated with testis cancer.
And we think that that's causal?
I don't know. It just worries me. Weed worries me.
Interesting, given that it otherwise seems, kind of benign. I personally can't stand this stuff, but I know so many people that use it so frequently that seem to have relatively few
it's an
effects.
Interesting phenomenon. It's medical marijuana, right?
Yeah.
So medical means safe. But I asked someone, I have a lot of pot growers in the Emerald City up in Northern California, and they have the artisanal stuff that wins awards and stuff. And it's like, which is worse for driving, being stoned or being drunk?
Undoubtedly being drunk.
Yeah. So it looks like reflexes, but like he said to me, Well, we tend to stop at stoplights and wait for them to turn.
Yeah, exactly.
When we're stoned, it's like
I'm not saying that one is. I'm not saying that they're both that driving stoned is good, but there are probably far fewer people that die at the hands of a stoned driver than a drunk driver.
Probably, and I think the signs, the LA story signs in LA, like if they have they have the lit up signs about open season traffic. Remember the movie LA Story? They do say now, stoned, drunk or stoned, watch out.
Yeah.
We're going to get you.
What do you think is the mechanism of action by which THC is having these negative fertility impacts?
Not sure. I don't think it's the mechanism. I don't think it's the root. So I don't think it's toking or edibles, but it might just be the chronic exposure. And I don't see, there's some evidence that THC acts like LH and binds the receptor and neutralizes and blocks it.
But blocks it from LH.
So you can get low testosterones, but it's not been that profound.
Mm-hmm. What about nicotine, either synthetically or in the form of tobacco?
Bad actor at high doses, I think, too.
Either one?
Yep.
Okay.
It's nicotine is the
nicotine
issue.
Per se. Yep.
Is the issue. And it doesn't last as long as THC. It does have, count motility effects and fertility effects. We think probably both of these are oxidants. It's the oxidants that do it. It's oxidizing things.
You mentioned diabetes earlier as part of your history and physical. What is it about diabetes? Is it the high levels of glucose? Is it the microvascular damage? Is it the inflammation that typically travels in parallel with it? Why is type 2 diabetes a risk factor for infertility?
Probably all of them. I don't think we know exactly, but I'd say that I diagnose diabetes in a lot of infertile men. I make the diagnosis.
What's the physical finding you're seeing in the testes that tells you, like, how an ophthalmologist will often make the diagnosis because they're looking into the eye?
So for me, it's usually their weight and their count motility are low. So I'm looking for a chronic exposure. And then they have polyuria or polydipsia or symptom like that, where they're drinking a lot and they're peeing a lot because the sugar is dragging it out. And you check their UA and it's full of sugar. And you said, and then some of them have an A1C that's a little pre-diabetic. But I think a lot of it is neurogenic too. Mm-hmm. They can develop an ED. They can develop, a third of type 2 diabetics have low testosterone. So that's a clue. And that's secondary. So you can clone it, you can bend them right back. But that's probably the common one, is the look, the sugars, and then the low T and the low sperm count. It's kind of a picture. It's a syndrome.
And then we've kind of talked about sleep and stress, obviously metabolic health in general. What are some of the other modifiable things that you see?
The most common is a varicocele.
Okay. Tell people what a varicocele is.
So you can develop varicose veins in your leg, which can need treatment. And this is the same thing in the scrotum, but it's not related. And it happens typically at puberty. You'll develop this. You won't know it sometimes unless it hurts. And it's a reflux of blood in the wrong direction. So the testicle drains to the kidney, which is uphill, and it wants to drain back down. The reason why it drains back down is because as a species, we stood up 500,000 years ago, maybe 750,000 years ago. And when you're an animal, your kidney and your testicle drains this way. There's no gravity. But when you stand up, you're now draining uphill. The system was never made for valves. And if you said to me, what's the reason our sperm counts are falling? I would say, we stood up as a species. Probably not a good idea for male fertility, because that blood that's supposed to be staying up there comes back down to the testicle, pulls around it like a hot bath is warmer, and it affects it. And usually the first sign is a testicle on that side, which is the left, usually is smaller than the right. So the physical exam will be a testicular discrepancy in size. That's the first thing you see. And then you feel above it and you feel a bag of worms.
But sorry, there are no valves in that vein?
Correct.
So what's the head it has to climb? That's got to be
30 centimeters.
So how is it doing that without a valve?
I don't know.
That's a pretty big distance to travel without a valve.
Maybe it does it at night. I don't know.
Yeah, interesting. Okay.
But there might be a few. But during puberty, with the growth spurt, those blow, and the angle of the renal vein there's a right angle.
Yep.
The right side has a natural valve off the vena cava, so it's kind of has to go around 270 degrees. So you don't reflux on the right. Left-sided lesion in most men. You can be perfectly fertile with it. If you look at statistically, 85% of men conceive naturally without varicoceles. 80% will conceive naturally about a year. So the curves are very similar, clinically maybe insignificant, but there's a difference, and it's statistical. But if you multiply that by millions of people, it becomes important.
And you'll figure that out easily on a physical exam.
The best way is easy. I don't order ultrasounds. If I can palpate it, then it's clinical.
And that's an office repair.
It's an outpatient surgery. It takes an hour. We do microsurgery.
Oh, it is? Okay.
Yeah.
So it's more involved than a vasectomy.
Yes, it is. And because if you're doing it at microsurgery at the level where you don't cut muscle, you want them to recover quicker. It's an involved area with lots of veins.
But he's not under general.
I use twilight sedation. Yeah.
Yep.
Okay. So I use it. Yeah. So that's the most common.
That's the most
40%.
common thing. Wow.
And most men are fertile, but they can be. So again, you look at the semen analysis as a poker hand, and you see count and motility being down, nothing else going on, and you see a varicocele, and it's implicated.
All right. Have we missed any other of the major?
Yeah, I'd say the major ones are varicocele, and then I would look for hormonal issues.
Yep.
So varicoceles may be 40, hormonal may be 10 or 15, genetics.
Yep. So they're non-modifiable now.
Right.
Okay. So you talked about a few of those already. What are some other ones on the genetic side? Yep.
The most common one is for zero sperm is Klinefelter, is XX chromosome. The most common one for low sperm count is Y chromosome deletions. This is an interesting area.
Right. What does that phenotype look like?
No phenotype. I mean, in general?
Yeah.
No phenotype. Normal looking.
A Y chromosome deletion male?
Yeah. Because it's only the long arm.
Yeah.
And it's only a couple of floors on the building.
Got it.
So there's regions that are missing.
I see. Okay.
So very interesting.
Oh, I'm sorry. I thought you meant a complete deletion of the Y chromosome.
Right, no,
Yeah. Got it. Partial.
So it's the long arm and it's deletions, regions. Delete.
Yep.
Ribosome deletion. Yeah, you're right. So Rene Riopera found that at MIT 20, 30 years ago now that the Y chromosome is a hall of mirrors. And in meiosis, every chromosome has a partner, except the Y and the X in a man. And so the Y plays with itself. It combines with itself instead of finding a partner. It has to do the dance too. And so it changes a lot. So it's very adaptable. It actually comes from the X through evolution. So there's a lot of X genes that are on the Y, and the Y we thought it was sort of a wasteland, maybe hairy ears and tooth decay and things like that. But now it's probably more important. So there are regions on the long arm of the Y. The short arm of the Y is very important. It has a gene called SRY, which makes you male. The SRY is the male sex-determining gene. If you have that gene, your phenotype will be male. If you don't have that gene, you're probably going to be female. It's complicated now, but that's sort of what it is. But the long arm has these genes that control fertility, and some of them, so typically we ordered in men with a low sperm count of below 5 million. So that would be a pretty common cause of a sperm count lower than 5 million. And I published a study that if you have a Y chromosome deletion and you have a varicocele, and they both cause low sperm counts, and you fix the varicocele, you're not going to improve because it's non-modifiable in all ways, right? It's who you are.
Yeah.
So, but if you didn't have the Y chromosome deletion and you fix the varicocele, you'll expect a good response. 2/3 will improve, 1/3 or more will conceive naturally. So you could take guys with low sperm counts and you can fix them or not, but the driver's genetics, right? And the phenotype in offspring is simply inherited as a Y chromosome deletion. It'll either be, I just had a couple from Texas actually. He had a Y chromosome deletion. He conceived with help of technology with a low sperm count. Sons have it, they have no sperm. So you can inherit the deletion, but it might increase. So you're going to get what your dad had, or it might be worse because mutations tend to get larger and large.
And until they try to conceive, they would never know this.
Right.
Everything else is normal.
Right. So then I would say there's environmental lifestyle things. So I think obesity is a big one.
Yep. And do you think that that's mostly propagated through the endocrine system then?
Yeah.
Yep.
And I think, that's a big one.
Oxidation. Yep.
In terms of the percent of sperm with the lifestyle issues. And then lousy diet is probably something that, and so obesity and diet, lifestyle, recreational drugs. What else do I review with them? Toxic exposures at work. So any smelly solvents, I'm really worrisome. Airport fuels, airline stuff, machine shop, oils, anything benzene derivatives, used to be pesticides and stuff like that, but they're pretty well controlled. So environmental exposures are kind of an unknown. I think viruses have a role.
Yep.
I saw you recently wrote about HPV, and I've been thinking about that for years because there are men. It used to be half the men who came in when I entered the field 30 years ago, we didn't know what was going on with them. But now it's probably like 10 or 20% with lifestyle issues and stuff like that. You can pretty much sort it out. It's not that unknown. But there are men who are like, what is going on here? He's a perfectly healthy guy. Practicing in California is incredible because everyone's so healthy, you have to look elsewhere. You have to ask other questions. And, when there's obesity, it's always the elephant in the room, but everyone is so healthy in eating. So I get to poke around places where no one else goes because I have to explain it and there's nowhere to go. But I did a study. So HPV is the most common. You wrote about that. What's the link? It's hard to know. There's herpes, very common. The STDs that we know about, like the 11 common beasts, chlamydia and gonorrhea and syphilis, those we know a little more about, and they're pretty obvious. But some of these, like trichomonas and stuff, are pretty subtle. And I was really concerned about this because one guy 20 years ago, now it's a professor at UCSF, he sent me a picture of my electron photograph of a sperm with a hexagonal herpes virus in it. And I don't even know if it was Photoshop, but it's like, there's this virus in a sperm. Like, yeah, it looks like there's a virus in that sperm. So you think that's what's causing it? I said, I don't know. But normally when you see infections as a cause, viral or bacterial, as a cause of semen analysis, you'll see pus cells. So you'll see what's called piospermia, leukocytospermia, the round cells we talked about. Yep. And the semen analysis will show up in higher numbers. They tend to be destructive and they tend to lower motility. So you tend to see a certain look to the semen analysis, volume, normal, count, normal. Motility is really low. A lot of the sperm are dead because they've been wiped out by these cytokines and all the white cells. And then maybe you'll find the pathogen somewhere, maybe not. But culturing mycoplasma, CMV, all these viruses. So Joe DeRisi, really bright guy at UCSF, won a MacArthur Award. He took my patient's semen, and he had it's back when microarrays were popular in the 2000s, and he had like 2,000 all mammalian viruses on his chip, everything. And we ran fertile guys and we ran infertile guys and looked at semen, not sperm. And 99% of the infertiles were positive for something, and 98% of the normals were positive for something. So ubiquitous was the word. And so it left us high and dry because you can't really do much with that. So it's out there. But I do agree with your assessment that the pathologic phenotypes, the worst ones, are probably doing something. The question is, how do we measure it? What do we look for? And the semen analysis, as I said earlier on, is a blunt instrument. It varies a lot. It's tough to do it, but I'd love whether we do genotyping is it in sperm? Probably not. I don't know. And when you look at HPV, it's probably one of those things that might be in the ejaculate after ejaculation. It might be coming from another fluid source and not in the sperm itself. So its effect would be post-ejaculation, which could still have a fertility effect, but it won't be probably as deep, right?
And what if, for example, a guy has prostatitis and the prostatic fluid has pus in it, then that could sabotage the whole thing. And
right. I mean, the problem with the male system is it's all through the same tube, right? Yep. So urine comes through that tube and semen comes through that tube. So you have to look for infections in the urinary tract in anything like that when you're doing fertility, because pus cells kill whatever they see.
Yeah.
So if your urine's infected, so that's a big deal.
Now, have you done work with intratesticular PRP and stem cells?
Just stem cells, but not PRP.
Okay.
Not a big fan.
Okay.
As a trained stem cell biologist and someone trying to make sperm from skin, and working with some of the best stem cell scientists in the world, I have a lot of respect for them, but it's not that simple. So we've had, people have injected bone marrow into, there's 560 offshore stem cell companies in the world that will take your money and do things like stick PRP in there. They'll stick bone marrow aspirates, fat in your testicle. And I'd say my experience has not been favorable. These are some of the toughest cases in the world, and they come to me after that, and I do my techniques and I don't find anything. And the trials aren't really real. Yep. They're kind of come here, we're going to do this, and then we're going to do a microdissection on your testicle. but they didn't have one beforehand. So the chance of it finding it even without that is X, and they're finding X. So, it's just not well done. And I have my patients investigate all that. And I say, you do the work, you tell me who you found, let me call them.
Yep.
So I'll let them be the workers, and then I'll call them and I'll say, hi, I was just wondering about, do you have any papers, or what's the science behind it? And they usually hang up, or, it's really interesting. But so far I'd say it's unfounded.
Yeah, I've had a very similar experience with a few of my friends and patients who have wanted me to talk with some of their stem cell docs. And
you're pretty evidence-based, Peter.
Well, yeah. And so I accept the fact that they're not going to have remarkable peer-reviewed data, but
I agree.
It is amazing at how few individuals can provide even one cell layer of scientific reasoning. And it's a topic I'd like to explore more deeply on the podcast. It's, my guess is there are some indications for where it makes sense.
I agree with that.
But boy, I'd like to figure it out without people wasting
I think there's a
so much money.
There's a there, but it's just not that easy.
Yeah. So what is let's just say for every 100 guys that walk in your office who are struggling with infertility, what percentage of them will be able to conceive, assuming they are able to fully comply with the prescriptions that you provide, be it lifestyle or pharmaceutical, for example, hormone modulation, et cetera, without requiring, and let's exclude the 40% varicoceles, because you're going to fix those guys and they're fine. So 100 people who don't have a varicocele, who don't have a genetic condition. I'm going to really simplify this. Okay, so these are 100 guys that presumably have showed up with some iatrogenic reason for infertility. How many of those guys are going to be able to conceive without resorting to IVF?
I would say that's the goal of my practice. And I would say the answer is most.
Wow.
But the caveat is, you got to tell me about the woman. Because I will defer.
No. So the point is,
I did a paper. This is the only data I can give you. And so I did a paper where I saw men for their infertility evaluation, got it done, and I thought they were fine. They had varicoceles and stuff, but their semen analysis was normal, and my investigation of their risks, lifestyle, everything was good. And I said, you're fine. You're cleared. No one's ever said that before. And they went home and they said, Turek couldn't figure out what was wrong with us. I said, it's not what I said. I don't do women. I don't, in my expertise, I'm saying something positive here, right? No one's ever said, most people would say, I'm not sure why you're not conceiving. I said, I'm pretty sure you're not the problem.
Yep.
Okay. Didn't get interpreted like that. That got me a little angry. So I did a study with USC, and I took these men that I cleared and I called them up a year later. And I said, what happened last year after Turek cleared you? I had a resident do this. And the answer was 65% had conceived naturally. Another 15 to 20% conceived with IUI or IVF. These women were 35 years old, year and a half infertility. They weren't going to wait around. Most conceptions occurred within 6 months. I didn't do anything for them. I didn't fix their varicoceles. I didn't touch them, and even medication. I just said, you're fine. So I published it as a lifestyle study. Not that I was right.
Mm-hmm.
And the idea was they probably made changes. They probably took a nutritional supplement. They probably timed their sex better. They probably got out of hot tubs and all that stuff. And they were taking pills. I have a list of what they did. I had a table in that paper that said that 65% natural pregnancy rate. That is higher than anything I can offer as a treatment that we have published on, right? So if you fix their varicocele, or you rarely get a 65% natural conception rate. So I had a table of all the published conception rates for the technologies that work, and I'm saying this is even better. So if that addresses your question, that's the only data I have.
What advice do you give a guy who comes in your practice? Maybe you don't see a lot of these guys, but let's say you get a guy who comes in and says, "Hey, look, I want to bank my sperm. I want to freeze my sperm." Now, presumably you'll get a lot of that if a guy's undergoing therapy for cancer or something like that. Is there anything a guy needs to know? And would you recommend a guy do that if he's 40, doesn't have a partner, but says, look, I want to have kids? And isn't there something to the idea that my sperm are better today than they will be in a decade?
It's a huge issue. Paternal age.
Yeah.
Paternal age and fertility, paternal age. So we can go there, but I don't place value judgments. I say good idea. I am a disclosure. I'm on a board of Legacy. I love their mission driven, the way they're mission driven. And so I like the fact they're going for military and exposed patients and this and that and VA. So, but I'm for that. I think it's the lowest hanging fruit in the field, especially obviously for cancer survivors and things. Like, I don't care what you think might happen with your cancer, I would still bank it. I started a nonprofit called Banking on the Future. 16-year-olds to 21-year-olds with cancer, we'll do it for you. We'll pay for it for 5 years. Just give us a sample, because it's so much harder afterwards, or not, right? So
you would advise any male that hasn't reproduced and who might want to, who's undergoing any chemotherapy for any cancer, just play it safe, bank.
For cancer, yes. Now, should anyone do it for any reason? Probably not. But again, I don't pass the judgment. If they're worried about something, then I'm worried. Then they should.
What paternal age do you worry about?
So, good question. And you look at like national guidelines for sperm donation. 40 is considered older paternal age. 50 for sure, right?
Mm-hmm.
If you look at risks to offspring, miscarriages, stillborns,
autism,
birth defects, things immediately related to conception, prematurity, those go up with paternal age. Then you look at birth defects when they're born; those go up, 1 to 2-fold. And then the worrisome ones are the single gene defects and the epigenetics, like psychiatric morbidity. So the autism, schizophrenia, dyslexia, bipolar disorder, potentially Alzheimer's in offspring. And they're not detectable with young. So big issues. So I've written a lot about that, published on it. I was actually having my second child at 50 when I was writing this thing. Should I be doing this? Writing a paper on all these risks called and with Alan Dichenko from University of Pittsburgh. But I think it's a hockey stick curve for risk to offspring.
And you think the inflection is 40 or 50?
I think it's more like 60.
Okay.
I think there's a slow linear increase in risk to offspring from 25 to 50 or 60, and then there's an inflection, and then there's the blade of the stick. And I think that's logarithmic. It's the same curve as women with chromosomal.
But they're shifted 20 years earlier or something like that.
So it's a shorter curve, but the same thing, 38 to 40 is kind of a point where things really ramp up with chromosomes. The men's stuff is not chromosomal. So if you take the curves together, they're different spans, same shape, but I think the female curve is on top of the male curve. This is not the same relative risk, right? So women, you go from 25 to 40, your chance of a miscarriage, though it's chromosomal, it goes up quite significantly after that, very significantly. And the consequence of women's issues with offspring-related health is basically miscarriage.
Yeah, and in many ways, it's almost easier to detect, right? Because So it's
been doing it for years.
It can be more dramatic, but it usually
is now prevented with pre-implantation genetic testing. Men are different. You can't detect these things. There's single gene mutations. This is the machinery is constantly working. It's getting old. The quality control of the process goes down, and little gene mutations get in there that are always being spun off in the heat of the engine, and they're not getting vetted. So the machinery is not doing a good job. So they're getting through, and they're not going to be lethal. They're going to be deleterious. So that's where you get things, and autism's a classic one, paternal age related. Looks like that's the biggest risk factor for it. And that worries me a lot, right? So I think it's interesting. The facts are that human evolution is entirely driven by sperm. Because eggs are just sitting there correcting the problem. It's entirely driven by sperm. And so 50 mutations a year, a generation usually gets spit out. It's in a Nature paper, probably between generations. And there's always mutations occurring in 14-year-old fathers, but it goes way up with 60-year-old fathers. So the rate of mutations goes way up with age, but it averages 50 over a reproductive life. And most of them, half of the mutations that we are throwing off as a species are not ears or hands or feet or height or things. It's all neurodevelopmental. It's like half neurodevelopmental.
Yeah.
So when you think about what we're seeing, the Martians from the 50s and the movies with big heads, that's kind of where we're headed. It's autism, dyslexia, bipolar disorder. These are neurodevelopmental, neurodegenerative issues. And why is that? Well, that's what's going on. I mean, that's where we're being stimulated. That's where we're being asked to evolve. Look at the last 30 years, right? Funny. One of the biggest investors in Salesforce said to me, I realized I was dyslexic when my son was born. And I said, really? He said, yeah, but you know what? It helped me be the man I am to realize that Salesforce is going to fly. Gave him the first 500,000, gave him the first million. They never took any more money. And he said, it let me focus. So autism is one of those diseases where you put out, you ignore a lot of input and you find the gift, and it's amazing. If you go down the rabbit hole of what they're good at, it's like their whole brain trust is there. So is that a disease or is that where we're headed?
I mean, I think it exists on a spectrum. I think anyone who's probably spent time with kids, using ASD as an example, boy, mild versions of it, the way it can be defined, because it really has 3 categories now in the DSM-5. I think the mildest version probably comes with more superpowers than limitations, or maybe equal amount, but clearly the more severe it gets, it's pretty debilitating. But this idea of paternal age,
we're calling it disease, though.
Yeah,
but maybe it's not disease. Maybe it's where we're headed. Maybe it's the future. Maybe the non sequiturs that come out of those brains. I mean, look at who's changing the world right now, at least in Silicon Valley.
Yeah. But again, I would argue most of those people would be in category, class 1, not class 3. What's the thaw success rate, right? So if a guy is 40, he goes ahead, he freezes and banks his sperm, assuming they were good to go in.
Right.
Are they very high probability of thawing correctly?
So when you freeze sperm, it's about a 200-year-old process, regularly used for about 75. I forgot who the Italian scientist was who froze sperm in snow and then thawed it, and it was alive a couple hundred years later. After Leeuwenhoek came up with a microscope, they found it was moving, and it was possible. So egg thawing is very new. Egg freezing and thawing is very new. This is very old. So everyone is thinking about sperm now because eggs are being frozen left and right. But this is much older technology, and the cell is much hardier than an egg. So it does a lot better typically. When you freeze it, it's the freezing process that kills sperm. Be from icicles on the inside.
Yes.
And then while it's frozen, there's usually no issue.
No issue.
And then there's another problem when you thaw it, rapid temperature shift. So that's where the kill rate comes from. In a good sample, half of it should survive.
Okay.
And so,
how much sperm would you tell a guy to bank if he has to do it, if it's the definitive samples for his life? So meaning he's 40 or he's about to undergo chemotherapy or some other exposure where he should just assume he will not have normal sperm again. What do you tell him?
So I usually say, depending on what technology you're going to use, but if your sperm count's normal, three ejaculates is 1 kid's worth of sperm with insemination technology where you would thaw it and then turkey baste it.
So 10 ejaculates for 3 shots on goal for 3 kids, potentially.
For 3 kids with low technology, but 10 ejaculates will give you most of China with IVF.
Got it. Oh, when you say low technology, you mean IUI or something like that? Right,
so there's 3 levels: sex, no tech, high tech is IVF, and then the middle is IUI.
Yep.
That's the stuff that's turkey basting. It's relatively straightforward, relatively cheap.
I see.
3 kids for that, but plenty of sperm for IVF.
So 3 ejaculates would be more than enough
if they're normal
for IVF.
Yeah. So the population you're talking about, maybe cancer survivors, half of those will not be normal. They're really looking at IVF.
Yep.
So they don't need that many.
Yep.
But I'd say 3 is a good number, but it's an insurance policy.
Yeah. Right? Okay. Well, Paul, this has been a super interesting tour through the world of male fertility. I can honestly say I knew very little about this coming in. Some of this stuff I understood pretty well, the hormone stuff, but boy, a lot of this stuff I had no idea. So I will be studying my notes from this. We're going to link to a lot of the stuff you've created. You've got a lot of great content out there, so we'll make sure people know where to find you.
Podcast too.
Tell me about that.
Well, we started a podcast last year because the blog of 15 years, and we're just doing timely topics, and it's me and my associate Rob Clyde, who's a director in Hollywood, and we take on Anthony Bourdain. We're going to be the Anthony Bourdain of men's health. We're going to just take on the topics, testosterone, et cetera, penis myths, and just talk about stuff that everyone is asking questions about, but no one's talking about. And like you, data-driven answers.
Okay. And what's it called?
Talk with Turek on all channels.
Got it. Okay. We'll make sure folks know that. You have a clinic that you run up and down the coast of California. So obviously we'll make sure folks know how to find you there. But presumably, I think we've given folks a roadmap for their local urologist as well if they're getting the workup. Basically, it sounds like if you're being worked up for fertility with your urologist and they're not going through the steps that we've described, maybe you should find somebody else.
Yes, I think it should be done. That's the first step. And there's a lot going on now that the biomarker concept, this relates a lot to your views on medicine 3.0. We now know, so the paper came out 2 days ago looking at longevity based on the semen analysis in Danish on the Rigshospitalet in Copenhagen. They looked at 74,000 men over 50 years and found that those guys with, say, normal semen quality live 3 years longer, all causes, than men with low sperm counts when they were younger. This was a single payer system, so they have all the data on it. It's very much a landmark study. So if you ask me what excites me about the field, I would say, as the author of the biomarker concept early in my career, I would say I'm really happy that we're scaring couples to realize that their fertility is a measure of their health. And now we have our foot in the door. If we can get a sperm count and get them in the office, we can actually tell them a little bit about their trajectory. And that's becoming more and more every day. And we've never had a chance to do preventative medicine with young men. So it's a men's health play in a big way because their partners are bringing them in, but who cares? They're in the office. What do you mean? Your father had prostate cancer when he was 50, someone had colon cancer, and so I have now an NP, Molly Jessup, who's medical. And it's like, okay, what's going on? Because there's metabolic stuff and you can pick up diabetes. And we have an opportunity here we've never had, ever, is to get men at younger ages. And I was a professor at UCSF for 15 years, endowed chair. I left. And I went to Yosan University, traditional Chinese medicine. I lecture there now. We had a conference last week. And I lectured, and I liked it because I thought Western medicine, maybe your view too, is too reactive. They're always trying to get men out of trouble or get patients out of trouble, but we're not thinking about getting them from unhealthy to healthy, which is the preventative aspect. We're just not very good at it. Your general surgery, every example you give is a guy who's did something bad, you get him back, whatever. But you got to think next step, like kidney stones, great. Urologists, we treat them all day. It's fun. It's endoscopic. It's lasers. It's shock waves. But what are we doing about that stone? I mean, how come we're not preventing these more? It's not on the radar. I go to Yosan University in traditional Chinese medicine, fabulous place, and it's all holistic. So I see patients who get referred by acupuncturists and they come in, their diet's under control, their stress is under control, they're doing acupuncture, they're sorted out. And what do I find? Varicoceles, because they don't find those. But the phenotype is totally different than the Western referral. And I think I've loved that because that's 3.0. That's medicine 3.0, which they're doing. They've been doing it for 4,000 years. And it's interesting how we don't give a lot of street cred to it. But in my view, much of we don't understand about fertility, certainly men, possibly women, is epigenetic. And the drivers of epigenetics, which are marks on the DNA, not DNA mutations, 50 DNA mutations a generation doesn't explain it. There's other stuff going on. Epigenetics is all lifestyle and diet driven. It's all lifestyle and diet driven. It's everything in your book.
Well, Paul, very interesting stuff. Thank you again for making the trip out here, and thanks for sharing your insights with us.
It's been great, yeah.
All right.
Thanks, Peter.